So today we are doing a deep dive into a massive stack of pathology and neurophysiology notes covering CNS demyelinating and dysmyelinating neuropathies.
It's a lot of material.
It is, yeah. And the mission for this deep dive is to pull apart the specific mechanisms that are driving things like multiple sclerosis, progressive multifocal leukoencephalopathy, acute disseminated encephalomyelitis, and a few critical osmotic and metabolic mimics too.
Yeah, we want to map out exactly why these systems fail at the cellular level. Not just what they are, but the exact mechanism of why they happen.
Right. Okay, let's unpack this, starting with the basic anatomy, the embryological origin of insulation itself, because, the cellular lineage really dictates everything about how these diseases manifest.
It really does.
Yeah.
So the central nervous system fundamentally forms from the neural tube during embryonic development.
Okay.
And within that neural tube, you have this innermost layer, the neuroepithelium, and it generates a lineage of precursor cells called glioblasts.
Glioblasts, right.
Yeah, and these glioblasts differentiate into various support cells. But the ones we are hyper-focused on today are the oligodendrocytes.
The oligodendrocytes, those are the big players here.
Exactly. These are the specialized cells that manufacture and maintain myelin exclusively within the central nervous system. So just the brain and the spinal cord.
And the divergence right there is what makes the pathology so incredibly specific, right?
Yeah.
Like, oligodendrocytes handle the CNS.
Right.
But the moment a nerve fiber exits the spinal cord and becomes part of the peripheral nervous system, the myelination is taken over by a completely distinct cellular lineage.
Yeah, the Schwann cells.
Exactly, Schwann cells, which come from the neural crest, not the neural tube.
Yes. And that is such a critical distinction. The demyelinating diseases in our source material today are laser focused on destroying the oligodendrocytes.
Just oligodendrocytes.
Just them. They leave the Schwann cells completely untouched, meaning your peripheral nerves are structurally spared.
That embryological boundary is just absolute, isn't it?
It really is. So when we see a patient with CNS demyelination, their peripheral reflexes, their peripheral nerve conduction velocities, they might remain entirely normal.
Yeah.
Even while their central tracts are being systematically degraded.
Wow.
Yeah. But to understand the impact of that degradation, we really have to look at the physics of how myelin actually facilitates electrical transmission.
Right. Before we break the wiring, we have to understand how it works. And the process is termed saltatory conduction.
Yes, saltatory conduction.
And the terminology there often creates this lingering misconception, I think, because saltatory comes from the Latin root saltare, which means to jump.
Right, to jump.
Yeah, and if you look at older diagrams, they literally draw the action potential leaping through the extracellular space, like from one gap in the myelin to the next.
I've seen those, yeah.
But reviewing the biophysics in these notes, that is mathematically impossible. Electricity doesn't jump through space like a spark plug in your brain.
No, definitely not. What's fascinating here is how myelin acts as a highly specialized dielectric material.
Okay, break that down for us.
So by wrapping tightly around the axon in these concentric layers, it drastically increases the transverse membrane resistance.
Meaning it stops leaks.
Exactly. It prevents the outward leak of positively charged ions.
Yeah.
And simultaneously, it increases the distance between the intracellular and extracellular fluids, which profoundly decreases the membrane capacitance.
So less capacitance means less charge actually needs to accumulate on the membrane to change the local voltage, right?
You've got it. So when an action potential fires, the electrical current flows passively and almost instantaneously down the axoplasm underneath that myelinated segment.
Right.
But because there is always some minuscule internal resistance, the signal eventually degrades. It needs to be actively regenerated.
And that regeneration happens exclusively at the nodes of Ranvier, right? Those tiny, unmyelinated gaps between the myelin sheaths.
Exactly. Those nodes are just densely packed with voltage-gated sodium channels.
So it's basically a repeater station.
Yeah, that's a perfect analogy. When the degraded passive current reaches the node, it's still strong enough to cross the threshold potential, triggering all those channels to open.
And boom, a massive influx of sodium.
Yes. A massive influx occurs, a new action potential is actively generated, and the cycle continues. It is this brilliant evolutionary adaptation that maximizes conduction velocity while minimizing the metabolic energy required.
Because the little na plus K plus ATPase pumps only have to work at the nose rather than along the entire length of the axon.
Exactly. It's incredibly efficient.
But the moment an oligodendrocyte is destroyed and that myelin is stripped away, that delicate biophysical balance just totally collapses.
It does. The exposed axonal membrane doesn't have the high density of sodium channels needed to propagate the signal continuously.
Right.
And without the myelin, the membrane capacitance skyrockets while the resistance plummets.
So the current just leaks out into the extracellular space.
Yep. The signal dissipates. By the time that passive current reaches the next node, it has degraded so severely that it fails to reach the threshold potential.
Meaning the voltage-gated sodium channels stay closed.
Exactly. They remain closed and you get a complete conduction block. The nerve fiber might be physically continuous, like it's not cut, but electrically, it is severed.
Wow. And the sources use a framework here that perfectly frames these downstream effects. They call it the CNS highway analogy.
I love that analogy.
Me too. If you map the nervous system as infrastructure, the cerebral cortex and deep gray matter are your starting cities. The dense white matter tracks those highly myelinated axons, those are the interstate highways.
Right.
And the target muscles or sensory organs, those are the destination cities. These demyelinating diseases are basically tearing up the asphalt on the interstate.
Exactly. The gray matter might be metabolically healthy, and the peripheral muscle fibers might be totally fine and fully innervated by their lower motor neurons. But the cargo never arrives.
So the pathology is entirely in transit.
Right. And framing it as an infrastructure failure sets the stage perfectly for multiple sclerosis. Multiple sclerosis, it's not just a generic illness where things slowly stop working. It is an exquisitely specific cascade of molecular events.
Yeah.
We really need to understand the physics of that city electrical grid, your central nervous system, and exactly how this friendly fire attack is staged, executed, and temporarily repaired.
And we're also going to dig into the evolutionary biology behind who gets this disease, which honestly contains some of the most surprising, counterintuitive medical insights you'll ever come across.
Oh, it really does.
Okay, let's unpack this. We have to start with the foundational science, right? The architecture of the grid itself. The central nervous system, meaning the brain and the spinal cord, relies on these long nerve axons to send signals.
Which are our copper wires in this analogy.
Right, and the thick protective rubber insulation wrapped around those wires is the myelin sheath. So, multiple sclerosis is defined as a chronic autoimmune inflammatory demyelinating disease.
The immune system is stripping the insulation.
Exactly. But the thing is, this isn't just a blind attack on all insulation in the body. It's incredibly targeted, isn't it?
It is remarkably, almost surgically targeted. And to understand this, we have to look at the specialized utility workers that actually manufacture this insulation.
Okay.
In the central nervous system, myelin is produced exclusively by a type of glial cell called an oligodendrocyte.
Oligodendrocyte. Okay, so oligo meaning few, dendro meaning branches, and cyte meaning cell. So literally a cell with a few branches.
Exactly. Just picture an octopus, but instead of 8 arms, it has maybe 15 or 20.
Oh, wow.
Yeah, and each of those arms reaches out, grabs a different nearby nerve axon, and wraps a thick sheet of a fatty substance, the myelin, around a segment of that wire. So one single oligodendrocyte can actually insulate multiple different axons simultaneously.
So they are the central utility workers. But if we leave the brain and spinal cord, like if we travel out into the peripheral nervous system, the nerves running down your arms into your fingers down to your toes, the workers change entirely.
They do. And this is a foundational concept. The peripheral nervous system does not use oligodendrocytes. It uses Schwann cells.
Okay, Schwann cells.
Now, Schwann cell is very different. It doesn't have multiple arms. The entire Schwann cell physically wraps its own body around one single segment of one single axon.
Oh, so it's a 1-to-1 ratio?
Right, it's a 1-to-1 ratio.
It just hugs the wire and literally becomes the insulation itself.
Precisely. And this difference isn't just cosmetic, it's developmental. I mean, oligodendrocytes originate from the neural tube, the neural ectoderm, during embryonic development.
Okay.
But Schwann cells originate from the neural crest. So they are biologically, genetically, and antigenically distinct from one another.
Which means they have different protein markers on their surface. They basically wear different uniforms.
Yes, exactly.
And this is why the immune system's attack in MS is so fascinating. The rogue security force has a highly specific hit list. They are looking for the uniform worn only by the oligodendrocytes.
Right, they absolutely hate central nervous system myelin, but they completely ignore the Schwann cells in the periphery.
It's just a phenomenal demonstration of immune specificity. I mean, the antibodies and T cells mediating multiple sclerosis will circulate right past a peripheral nerve in your arm and do absolutely nothing to it.
So if you are looking at a clinical case or evaluating a patient and they have demyelination happening in their peripheral nervous system.
Like maybe they are experiencing ascending weakness starting in their legs and moving upward.
Exactly.
Yeah.
Then you are absolutely not looking at multiple sclerosis.
Yeah.
You're likely looking at Guillain-Barré syndrome, which is an autoimmune attack specifically targeting peripheral myelin, the Schwann cells.
Got it.
In MS, the destruction is confined strictly behind the walls of the central nervous system.
Okay, so the city grid's central insulation is being ripped away. We know it is being destroyed. But let's dive into the physics of why that insulation matters so much. Because there is a very persistent myth here that we need to bust.
Ah, yes. The jumping myth.
Yes. When we talk about how an electrical signal travels down a myelinated nerve, we use the term saltatory conduction. The Latin root is saltare, which means to leap or to jump.
Right.
And I think because of that word, a lot of people picture the electricity literally jumping through the fluid like a spark plug, sparking over the insulated parts from one bare spot to the next. But that's not right, is it?
No, it's an incredibly common visual misconception. We see it drawn that way in simplified diagrams all the time, arrows bouncing over the myelin like a frog on lily pads.
Yeah, exactly.
But physically, biologically, that is completely impossible. The electricity does not leave the cell, jump through the extracellular matrix, and land back on the nerve.
So what is actually happening? How does the signal actually travel?
To grasp saltatory conduction, we have to look at the anatomy of the wrapped wire. The myelin sheath isn't one continuous unbroken tube of rubber. It's laid down in segments, like lengths of sausage.
Okay.
Between each segment of myelin, there is a microscopic, uninsulated gap in the axon membrane. These gaps are called the nodes of Ranvier.
Right, the bare spots on the wire where the copper is just exposed.
These nodes of Ranvier are absolutely packed with voltage-gated sodium channels. When the electrical signal, the action potential, hits this bare spot, these channels fly open, sodium rushes into the cell, and the signal is actively regenerated. It's like a booster station along the railway.
But the booster stations are separated by these long insulated stretches of myelin, which are called the internodes, right?
Yes.
And there are no sodium channels under the myelin. So how does the signal get from one booster station to the next without jumping?
It travels inside the axon, underneath the myelin, as a passive electrical current. And it does so incredibly fast.
So it's an internal spread.
Exactly. And to understand why it's so fast, we have to look at two fundamental properties of electrical physics resistance and capacitance.
Oh, let's break those down because this is where the genius of evolutionary engineering really shines. Let's start with resistance.
Okay, so think of the axon membrane as a garden hose, and the electrical current is the water pressure inside. A naked, unmyelinated nerve membrane is actually quite leaky.
Okay, it has holes in it.
Right. Positive ions tend to leak out across the membrane as the current flows down the tube. Because of this leakage, the signal loses strength very quickly, meaning the nerve has to constantly stop and actively regenerate the signal every fraction of a millimeter.
That sounds extremely slow.
It is extremely slow, and it requires massive amounts of metabolic energy.
But when the oligodendrocyte wraps its fatty myelin arms around that hose,
It acts as an incredibly thick waterproof tape. Myelin drastically increases the transverse membrane resistance. It plugs the leaks.
Oh, I see.
The charged ions can no longer escape across the membrane. So all of that electrical pressure is forced to rapidly push forward, straight down the center of the axon, toward the next node of Ranvier.
So it's basically forcing the current into a high-speed underground subway tunnel. That's resistance. But what about capacitance? Because that's a slightly trickier physics concept.
It is. Capacitance is essentially the ability of a system to store an electrical charge. In a naked nerve, the cell membrane is very thin. You have negative charges sitting on the inside of the membrane and positive charges sitting directly on the outside, separated only by a few nanometers.
Okay.
Because they're so close, they're highly attracted to each other. They stick to the membrane.
Like magnets holding tight through a thin piece of paper.
Precisely. And because they're stuck there, before the nerve can send a signal down the line, it first has to spend time and energy unsticking all those charges, filling up that capacitor,
Right.
before the voltage can actually change and propagate. It's a huge drag on the system speed.
So how does myelin fix the drag?
Well, myelin is thick. We're talking up to 100 layers of dense lipid membrane wrapped around the axon. This physically pushes the extracellular positive charges much, much further away from the intracellular negative charges.
You're separating the magnets. You're putting a thick textbook between them instead of just a piece of paper.
Exactly. The attraction drops to almost zero. The membrane capacitance is drastically decreased.
Right.
The charges are no longer sticking to the walls, which means the electrical current doesn't have to waste time charging up the membrane.
Sure.
The voltage change simply sweeps down the interior of the axon with almost zero friction.
Wow. So you have low capacitance, meaning no drag, and high resistance, meaning no leakage. The current just sweeps under the myelin at blistering speed, hits the bare node of Ranvier, triggers a massive influx of sodium to boost the signal, and sweeps forward again under the next myelin segment.
Yes.
It looks like the signal is teleporting or jumping from node to node, hence the word saltatory, but it's really just this incredibly efficient, high-speed underground transit system.
Which brings us back to the pathology of multiple sclerosis. If you take that exquisite physical system and you introduce the rogue security force, if the immune system strips off that myelin, what happens to the physics of the wire?
It's a total logistical collapse.
Absolute collapse. The moment the myelin is gone, the high resistance is gone, the leaks are back.
Right.
The low capacitance is gone, the drag is back. When the signal hits the damaged, demyelinated area, the electrical current just dissipates out into the surrounding tissue.
The pressure in the hose drops to zero.
And by the time whatever tiny trickle of current is left finally reaches the next node of Ranvier, it's too weak. It doesn't have the voltage required to trigger those sodium channels. The threshold is just not met.
So the signal doesn't just slow down, it literally dies. A complete conduction block.
The action potential fails. The wire is physically still there, at least in the early stages of the disease, but the electricity cannot make the trip. And this conduction block is exactly what translates into the sudden neurologic deficits the patient experiences. If that wire was carrying visual data from the eye, the patient goes blind. If it was carrying motor commands to the leg becomes paralyzed. The physical infrastructure is compromised.
Okay, so the insulation is gone, the physics are broken, the current is leaking. But we need to look closer at the actual perpetrators. We need to identify these rogue union workers who are ripping the city grid apart.
Right, the immune cells.
Yeah, let's pivot to the immunology, this friendly fire attack. Because this isn't a blunt force trauma, this is a highly orchestrated infiltration.
It is. If we zoom out and look at the broader classifications of immunology, multiple sclerosis is primarily driven by a type IV hypersensitivity reaction.
Now, type IV means it is cell-mediated, specifically T-cell mediated. This isn't like a type I hypersensitivity, like a peanut allergy or asthma, which happens in minutes and is driven by histamine and IgE antibodies.
Not at all.
Type IV is delayed, and it's driven by the cellular infantry.
It is the heavy machinery of the immune system. The process initiates with peripheral autoreactive T cells. We all have immune cells that are born with the potential to attack our own tissues, but normally the thymus gland weeds them out.
It deletes them before they can enter the bloodstream. That's central tolerance.
But in MS, a few of these T cells escape that deletion. They lose their tolerance. Specifically, we are looking at CD4 positive helper T cells, heavily leaning into the Th1 and Th17 subsets.
But here's the logistical problem for these rogue T cells. They are circulating out in the peripheral blood. The central nervous system is a fortress.
A literal fortress.
It is protected by the blood-brain barrier.
Yeah.
The endothelial cells that line the blood vessels in the brain are fused together with these incredibly complex, tight junctions. Plus, you have the astrocytes wrapping their end feet all around the vessels just to double seal the gates.
Right.
Nothing gets in without a VIP pass. So how do these rogue commanders breach the castle?
Well, under normal circumstances, you're right, they bounce right off. The blood-brain barrier is practically impenetrable to circulating immune cells.
Okay.
But these autoreactive T cells, through a complex mix of genetic and environmental triggers we'll discuss later, become activated. When a T cell is activated, its genetic expression changes. It starts manufacturing and displaying specific adhesion molecules on its outer surface.
Like Velcro.
Exactly like Velcro. The key players here are called integrins, specifically alpha-4 beta-1 integrin, also known as VLA4. Now, simultaneously, there is some sort of low-level initiating inflammatory signal coming from the blood vessels of the central nervous system, causing the endothelial cells there to express a receptor called VTAM1.
Okay, so the T cell has the alpha-4 integrin Velcro, and the blood vessel wall puts up the VCAM1 Velcro pad.
When they circulate through the brain, they stick. This process is called margination and rolling. The T cells literally pull themselves out of the fast-flowing bloodstream and lock onto the vessel walls.
Wow.
But sticking to the wall isn't enough. They have to get through it.
They have to pry the doors open.
And they do this chemically. The T cells release enzymes called matrix metalloproteinases, or MMPs. These enzymes act like molecular blowtorches.
Blowtorches?
Yeah, they digest the extracellular matrix and physically break down the tight junctions holding the blood-brain barrier together. The doors are blasted open, the barrier becomes highly permeable, and the T cells undergo diapetesis. They squeeze through the vessel wall and migrate directly into the central nervous system white matter.
The breach is successful. The rogue commanders are inside the fortress. Now, T cells, especially these CD4 helper T cells, they don't usually do the dory work themselves, do they? They're the generals. What do they do once they set up their command post inside the brain?
They start issuing orders. They broadcast chemical signals to recruit the rest of the army. The Th1 subset of T cells starts releasing massive amounts of inflammatory cytokines, particularly interferon gamma and tumor necrosis factor alpha. Meanwhile, the Th17 cells release interleukin-17, which is incredibly potent at recruiting even more inflammatory cells and further degrading the blood-brain barrier so more troops can flood in.
So they're calling in the demolition crew. Who actually wields the sledgehammers? Who physically rips the myelin off the wire?
Well, there are CD8 positive cytotoxic T cells present that can cause direct damage. But the vast majority of the physical destruction, the heavy lifting of demyelination, is executed by macrophages.
Right.
These are the monocytes that follow the T cells in from the bloodstream alongside microglia, which are the resident, built-in macrophages that already live permanently inside the brain.
So the microglia and the peripheral macrophages get activated by the interferon gamma. They converge on the oligodendrocyte and they just start chewing it up. Do they use a specific weapon?
They employ several destructive mechanisms. First, they engage in phagocytosis, literally engulfing and eating the myelin.
Oh, wow.
But they also release vast quantities of reactive oxygen species and nitric oxide.
Free radical.
Yes. Myelin is essentially a massive, dense sheet of lipids fats. Reactive oxygen species are highly unstable molecules missing an electron. They violently steal electrons from the lipids in the myelin sheath, a process called lipid peroxidation.
So it's breaking it down chemically.
It literally rusts and dissolves the fatty insulation right off the axon. Furthermore, they release proteases that specifically target and digest the proteins embedded in the myelin.
And what are those specific proteins? Like, what is the actual molecular target the immune system thinks is an invader?
The exact autoantigen isn't universally identical in every patient, which adds to the disease's complexity. However, decades of research have identified several major targets. The most famous is myelin basic protein, or MBP.
Okay.
There is also proteolipid protein, PLP, and myelin oligodendrocyte glycoprotein, or MOG. The macrophages recognize these specific proteins as foreign threats and relentlessly attack them.
Okay, here's where I have to throw a massive wrench into this explanation. We just spent all this time outlining how this is a type 5 face cell-mediated disease. The T cells breach the wall, the T cells call in the macrophages dissolve the lipid membrane.
Right.
But wait, I'm looking at the gold standard diagnostic criteria for MS-DIS. And when a neurologist does a lumbar puncture, a spinal tap, to look at the cerebrospinal fluid, they aren't primarily looking for T cells.
No, they aren't.
They are looking for antibodies. Specifically IgG antibodies.
I absolutely are.
But antibodies are produced by B cells. That's a humoral immune response, not a cell-mediated one. If T cells are the sole rogue commanders here, why are B cells leaving their fingerprints all over the crime scene? Like, why do we care so much about B cell antibodies if this is a type IV disease?
This is a brilliant observation, and it highlights a major paradigm shift in how we understand multiple sclerosis. For decades, the dogma was exactly as we just described. MS is a T cell disease. End of story. But that dogma was incomplete. While the primary initiating breach is indeed driven by T cells, the B cells are not just innocent bystanders. They are massive, critical contributors to the ongoing pathology. The T cells blast the blood-brain barrier open, but the B cells eagerly follow them inside.
And what do the B cells do once they get into the central nervous system?
They establish permanent residency. B cells migrate into the cerebrospinal fluid and the brain tissue, and under the influence of the inflammatory environment, they differentiate into plasma cells.
Okay.
Plasma cells are essentially microscopic antibody factories. They set up camp right there behind the blood-brain barrier and begin pumping out immense quantities of IgG antibodies.
So they are manufacturing the weapons locally.
Exactly. This is called intrathecal antibody production. Intra meaning inside the thalica, referring to the protective sheath of the central nervous system. The antibodies are being synthesized locally inside the fluid of the nervous system, not out in the general peripheral bloodstream.
And how do we actually see that on a diagnostic test? What is the physical evidence?
When we collect that cerebrospinal fluid via lumbar puncture, we analyze it using a technique called gel electrophoresis. We apply an electrical current to a gel matrix, which forces the proteins in the fluid to separate based on their size and electrical charge.
Because different proteins migrate at different speeds through the gel.
Precisely. In a healthy person, if you look at the region of the gel where IgG antibodies migrate, it's just a faint, diffuse smear. There's a little bit of background immune activity, but no single clone of B cells is dominating.
Right.
But in a patient with MS, those local plasma cells are churning out massive amounts of a few specific types of IgG antibodies targeted against the myelin.
So instead of a diffuse smear, those specific identical antibodies all clump together in the exact same spot on the gel.
And they form distinct, visually striking, dark bands. We call them oligoclonal bands. Oligo meaning a few, clonal meaning they come from a few specific clones of hyperactive B cells.
Oh, that makes sense.
Finding these oligoclonal bands in the cerebrospinal fluid, while not finding them in the patient's peripheral blood serum, is an absolute hallmark of multiple sclerosis. It proves definitively that there is a localized, rogue B-cell inflammatory party happening exclusively inside the central nervous system.
The T cells kick the door down, but the B cells move into the living room, set up a factory, and start throwing this highly destructive, localized inflammatory party.
It's incredibly destructive synergy. The B cells don't just produce antibodies, they also act as antigen-presenting cells, feeding pieces of the destroyed myelin back to the T cells, throwing gasoline on the fire.
Right.
In fact, some of our most powerful modern therapeutics for MS, which we'll discuss later, specifically target and assassinate B cells. And their massive success in halting the disease proves just how central the B cell is to the pathophysiology of MS.
Okay, so we have a deep understanding of the mechanics of the breach and the cellular players involved. We know how they fight, but we need to map the battlefield. Where exactly are these immune cells setting up their demolition sites? Let's trace the anatomy of an attack. The central nervous system is broadly divided into two types of tissue, gray matter and white matter.
And the geography of the disease is highly specific. MS is overwhelmingly a disease of the white matter, while largely, though not entirely, sparing the gray matter, especially in the early relapsing phases.
Just to anchor us, let's define those zones. Gray matter is where the actual bodies of the neurons live. The nuclei, the dendrites, the main computer processors of the brain, the cortex on the outer surface of the brain is gray matter.
And white matter consists of the incredibly dense tracts of axons connecting those processors. It is white because it is packed full of that fatty, lipid-rich myelin insulation.
Makes sense.
Since the autoimmune response is specifically hunting myelin and oligodendrocytes, it naturally congregates where the prey is thickest. The white matter tracks deep inside the brain and down the spinal cord.
And there is one highly specific location, a classic radiological finding, that every single medical student learns on day one of neurology: the Dawson fingers. Let's break this down because the mechanism behind it is fascinating. When you look at an MRI of an MS patient, specifically a sagittal or axial view of the brain, you often see these bright white oval-shaped lesions, and they are clustered right around the lateral ventricles, the fluid-filled open spaces deep in the center of the brain. They look literally like glowing, elongated fingers radiating outward from the ventricles.
It is a striking and highly characteristic visual pattern.
Now, when I first read about this, my immediate assumption was purely spatial. I thought, okay, the immune cells must be swimming in the cerebrospinal fluid inside the ventricles, and they are attacking the walls of the ventricles directly, causing these lesions right on the edge. Is that what's happening? Are they attacking the ventricular lining?
It's a very logical, intuitive deduction, but it is actually a major misconception. The immune system is completely ignoring the ependymal cells that line the wall of the ventricles. Yeah, the geographic location of those lesions has absolutely nothing to do with the ventricles themselves.
Then why are they always clustered right there, pointing away from the center?
It all comes down to the microscopic vascular plumbing of the brain. The deep, dense white matter immediately surrounding the ventricles. The paraventricular white matter is heavily populated by a very specific set of blood vessels called the small medullary veins.
Okay.
These tiny veins drain blood away from the deep brain tissue, and anatomically, They run perpendicular to the walls of the ventricles. They radiate outward.
Like the spokes on a bicycle wheel extending out from the center hub.
Exactly like spokes. Now, remember the breach we discussed? The T cells sticking to the endothelial cells and using MMPs to melt through the blood-brain barrier.
Yes, the blue torches.
That process does not happen randomly everywhere. The hemodynamic forces and the specific expression of adhesion molecules make the postcapillary venules, these exact small medullary veins, the absolute prime real estate for the immune cells to cross over.
Ah, so the T cells aren't coming from the ventricular fluid. They are leaking out of these specific little veins.
Yes, they exit the vein and accumulate in the tiny anatomical gap immediately surrounding the blood vessel known as the peривascular space or the Virchow-Robin space. Once they're sitting right on the outside of the pipe, they initiate the inflammatory cascade. The macrophages arrive and just start chewing up the myelin radially outward from the vein.
They just follow the highway.
Exactly. The inflammation and the resulting demyelination simply track along the physical anatomical path of the vein. Since the veins are shaped like long spokes radiating perpendicular to the ventricles, the resulting scar, the demyelinated plaque, forms a long oval shape tracing that exact path, hence Dawson fingers. Ultimately, MS is a perivenular inflammatory disease. The lesions map the veins, not the ventricles.
That reframes the entire visual perfectly. You aren't looking at ventricular damage, you're looking at the glowing footprints of the venous highways. Now, because all of this damage is locked firmly inside the central nervous system, the brain and the spinal cord that strictly dictates the physical symptoms the patient will experience, when we talk about motor weakness in MS, we are talking exclusively about upper motor neuron disease, or UMN signs.
Correct. And the distinction between upper motor neuron and lower motor neuron lesions is the absolute bedrock of localizing neurological disease. Because MS is restricted to the CNS, the peripheral nerves are spared.
Let's explain the chain of command here. The upper motor neurons are the big boss cells sitting in the motor cortex of the brain. Their axons travel all the way down the spinal cord through the white matter tracts called the corticospinal or pyramidal tracts. Eventually, they synapse with the lower motor neurons, which are the middle managers sitting in the anterior horn of the spinal cord.
Right.
The lower motor neurons then send their axons out to the actual muscles to tell them to contract.
That's the chain. Now, what does the big boss, the upper motor neuron, actually say to the lower motor neuron? Most people assume it just says fire.
Sure.
But actually, a massive part of the UMN's job is inhibition. It is constantly yelling at the lower motor neuron to calm down, to maintain a smooth, steady baseline tone, and not to overreact to every little stretch or stimulus.
It's essentially holding back the leash on an overly aggressive guard dog.
A perfect analogy. Now, in MS, the immune system strips the myelin off those upper motor neuron tracts in the spinal cord. The signal is blocked, the leash is cut. The lower motor neuron suddenly loses all of its cortical inhibition.
The guard dog is let off the leash. What happens to the muscle?
The lower motor neuron becomes hyperactive. Specifically, the gamma motor neurons that control muscle spindle sensitivity go into overdrive. The muscles become incredibly tight and rigid. Clinically, we call this spasticity. The patient experiences a dramatic increase in muscle tone. Furthermore, when you tap their knee with a reflex hammer, which is a simple stretch reflex, the uninhibited lower motor neuron panics and fires back with massive force. You see hyperreflexia, the leg kicks out aggressively, sometimes repeatedly, which is called clonus.
And this is also where we see the famous Babinski sign.
Yes. Normally, if you take a hard object and stroke the bottom of an adult's foot from the heel to the toes will naturally curl downward in a flexor plantar response. The upper motor neurons ensure this normal, mature reflex. But if you cut the UMN pathways with an MS plaque, the reflex reverts to an infantile state. Stroking the foot causes the big toe to violently point upward, or dorsiflex, while the other toes fan out. A positive Babinski sign is a glaring red flag for central nervous system damage.
And just as importantly, what will we not see? Because knowing what is absent is just as vital for diagnosis.
Because the lower motor neurons themselves and their peripheral axons wrapped in Schwann cells are perfectly intact and healthy, you'll generally not see severe, rapid muscle wasting or profound atrophy. The muscle is still getting plenty of electrical stimulation. In fact, it's getting too much. And you will not see fasciculations, which are those continuous, tiny, rippling muscle twitches caused by a dying lower motor neuron spontaneously misfiring.
So if a patient has spasticity, hyperreflexia, and an upgoing toe, We are in the central nervous system. We are looking at an MS profile. But if they have flaccid, floppy muscles, profound wasting and twitching, the problem is peripheral, and MS drops off the differential diagnosis.
The system is exquisitely specific, and the physical exam tells you exactly where the lesion lives.
Which brings us perfectly into the clinical picture, because we just described the motor pathways, but these Dawson fingers, these inflammatory plaques, can form literally anywhere along the thousands of miles of CNS white matter. The visual pathways, the brainstem, the cerebellum, the sensory columns in the spine. Because of that, this disease is the ultimate chameleon. It looks different in almost every single patient.
It is notoriously heterogeneous. A patient could present with numbness in their left foot, or blindness in their right eye, or an inability to swallow, or severe vertigo. But amidst all that chaos, there is a unifying framework for diagnosing MS, and it revolves around a core concept: dissemination in time and space.
Dissemination in time and space. This is the bedrock of the McDonald criteria. Let's define those.
Dissemination in space means that the immune system is causing damage in at least two entirely different physical locations within the central nervous system: an inflammatory plaque in the optic nerve and a separate plaque in the cervical spinal cord. They are spatially disseminated.
And dissemination in time means these attacks didn't just happen all at once from a single toxic exposure or a single stroke.
Exactly. The autoimmune attacks occur in distinct, separate clinical episodes, separated by months or even years. The immune system flares up, causes a lesion, calms down, and then a year later flares up again in a new location. Different space, different time.
So let's walk through some of the classic, highly tested AHA symptoms. The specific clinical presentations that, when a doctor sees them in a young patient, alarm bells for demyelination should immediately start ringing. You just mentioned the optic nerve. I was reading the literature, and it seems optic neuritis is incredibly common as the very first presenting symptom of MS.
It is often the herald symptom. The optic nerve, cranial nerve 2A, is essentially a direct extension of the brain itself. It's heavily myelinated by oligodendrocytes, making it a prime, highly vulnerable target. When a plaque forms on the optic nerve, the patient typically experiences rapid monocular vision loss.
Meaning just one eye suddenly goes blurry, or they lose a massive chunk of their visual field, usually right in the center. And it hurts, right?
It is characteristically painful, especially with eye movement. The inflammation causes the nerve and its surrounding sheath to physically swell. The optic nerve has to pass through a very tight bony canal to get from the eye to the brain.
Oh, that makes sense.
Yeah, when it swells, it gets compressed, and every time the patient looks left or right, they stretch that inflamed, swollen nerve, causing a sharp aching pain.
Now, if a patient comes in with this, the doctor will likely perform the swinging flashlight test to look for something called an afferent pupillary defect, or a Marcus gun pupil.
I've read about this, and it's described as a paradoxical dilation. Can we break down the actual physics and anatomy of what is happening during this test? Because the eye doesn't actually dilate in response to light, does it? That's a vital clarification. No eye will actively dilate because you shine a light in it. The paradox is relative. Let's trace the wiring. The optic nerve is the afferent limb. It carries the sensory signal of light from the retina into the brain.
Okay.
The brain receives that signal at the Edinger-Westphal nucleus and sends a motor command back out via cranial nerve III, the oculomotor nerve. That is the efferent limb, which tells the pupillary sphincter muscle to constrict.
Right.
And crucially, this is a consensual reflex. If you shine a light in one eye, the brain tells both pupils to constrict equally.
So under normal circumstances, both pupils are tiny.
Now imagine the patient has optic neuritis in their left eye. The left optic nerve is demyelinated. The signal is blocked.
The afferent limb is broken.
The doctor swings the flashlight into the healthy right eye. The right optic nerve sends a strong signal to the brain, lots of light. The brain tells both pupils to constrict tightly. Everything is working fine. But then the doctor rapidly swings the flashlight over to the inflamed, demyelinated left eye.
The left eye receives the light, but because the myelin is gone, the signal leaks out. The current drops.
The left optic nerve sends a very weak, degraded signal to the brain. The brain registers this massive drop in signal and thinks, oh, the room just got dark, I need to let more light in. So the brain withdraws its constriction command.
And both pupils relax and widen.
Exactly. As the doctor swings the light from the healthy eye to the sick eye, they watch the pupil in the sick eye actually get wider, appearing to dilate, despite the flashlight shining directly into it. It is dilating because the brain doesn't realize how bright the light actually is.
Wow.
That is the afferent pupillary defect. It physically proves that the sensory wire is severed or severely delayed.
That is an incredible piece of diagnostic logic. Just by watching the pupil, you prove the presence of demyelination behind the eye. So what does this all mean? Okay, let's move slightly deeper into the brainstem. There is a symptom called internuclear ophthalmoplegia, or INO. I have always found this entirely fascinating. How do the eyes literally stop communicating with each other?
This is perhaps one of the most brilliant pieces of neuroanatomy you will ever study. It involves the complex synchronization of eye movements. If you want to look to your left, you can't just move one eye. Your left eye has to look outward, away from your nose, which is called abduction, and your right eye has to look inward, toward your nose, which is called adduction.
And they have to do this at the exact same millisecond with the exact same speed, or you would see double vision immediately.
Right. They must be perfectly tethered together. But the muscles controlling these movements are driven by two entirely different cranial nerves in two different parts of the brainstem. Abduction, looking out, is controlled by cranial nerve 6, the abducens nerve in the pons. Adduction, looking in, is controlled by cranial nerve the 3rd, the oculomotor nerve in the midbrain.
So you have two different control centers that have to fire simultaneously. How do they actually coordinate?
There's a highly specialized, heavily myelinated copper wire that connects the abducens nucleus directly to the oculomotor nucleus. It acts as the synchronization cable. This tract is called the medial longitudinal fasciculus, or the MLF.
And as we know, MS absolutely loves to destroy heavily myelinated tracts.
It does. When an inflammatory plaque hits the brainstem and demyelinates the MLF, that synchronization cable is cut. So imagine a patient with a lesion in their right MLF. The doctor asks them to look sharply to the left. The brain sends the command to the left abducens nerve. The left eye looks outward, abducts, perfectly fine.
But the left abducens nerve tries to send the synchronization signal up the MLF to tell the right eye to come along.
And the signal hits the demyelinated plaque and dies. A conduction block. The right oculomotor nerve never gets the message. The right eye fails to adduct. It just sits there, looking straight ahead, paralyzed in the midline, while the left eye is staring off to the side.
That alone is wild. But what happens to the left eye, the one that successfully looked outward? Doesn't it do something weird too?
It does. The brain is constantly monitoring proprioception and visual input. It instantly realizes that the eyes are mismatched and the patient is experiencing double vision. The brain panics. It desperately tries to drive the left eye even harder or correct the position, resulting in the abducting eye, the left eye rapidly jerking back and forth horizontally. We call this abducting nystagmus.
So one eye refuses to move inward, and the other eye shakes violently. And if a clinician sees that specific mismatch, they know instantly, without even needing an MRI, exactly where the lesion is.
It is a highly localizing sign. If you see bilateral INO, meaning the MLF is cut on both sides in a young adult patient, it is almost pathognomonic for multiple sclerosis. It is one of the most reliable clinical indicators we have.
Let's travel down into the spinal cord. There is a classic sensory phenomenon called the Lhermitte sign. What is happening there?
The Lhermitte sign occurs when an MS plaque forms in the cervical spinal cord, specifically in the dorsal columns, which are the heavily myelinated tracts carrying vibration and proprioception sensory data up to the brain.
So the insulation is stripped off the back of the spinal cord in the neck.
Yes. Now, a demyelinated nerve is highly unstable. It's essentially a raw, exposed, frayed wire. It becomes exquisitely mechanosensitive, meaning physical stretching can cause it to spontaneously misfire.
Oh, wow.
When a patient with a cervical plaque flexes their neck, tucking their chin tightly to their chest, the spinal cord is physically pulled and stretched over the bones of the cervical spine.
They literally stretch the frayed wire.
And the result is a sudden, massive, spontaneous volley of action potentials. The patient experiences this as a transient, violent, electric shock-like sensation that originates in the neck and shoots rapidly down their spine, often radiating out into their arms or legs. It only lasts for a second, but it is deeply unsettling.
It's the literal biological equivalent of bending a broken power cord and getting a spark. Okay, what about the Charcot neurologic triad? I want to clarify right up front. We are not talking about Charcot's triad for cholangitis in the liver, which is fever, jaundice, and upper quadrant pain. This is completely different.
Yes. Jean-Martin Charcot was a very busy neurologist and pathologist, and his name is on many things. The Charcot triad in MS points to widespread demyelination, particularly involving the intricate feedback loops of the cerebellum. The cerebellum is the brain's coordination center. It ensures our movements are smooth, targeted, and precise.
And the triad consists of 3 specific signs of cerebellar failure: scanning speech, intention tremor, and nystagmus. Let's break those down. What does scanning speech sound like?
Scanning speech, sometimes called explosive or ataxic speech, sounds almost robotic or disjointed. Normal human speech requires the incredibly rapid microscopic coordination of the diaphragm, the larynx, the tongue, and the lips.
Right.
The cerebellum orchestrates that. When the cerebellar tracts are demyelinated, that coordination fails. The patient speaks with unnatural, prolonged pauses between syllables. The emphasis on words becomes erratic and unpredictable. I'm going to the store. The fluidity is lost.
And an intention tremor. This is a massive point of confusion because people hear tremor and immediately think of Parkinson's disease. But this is fundamentally different mechanically, isn't it?
Fundamentally different. A Parkinsonian tremor is a resting tremor. It is caused by a failure in the basal ganglia. The patient's hand shakes when it is resting in their lap doing nothing. But the moment they reach for something, the tremor often improves or stops.
Okay.
An intention tremor in MS is the exact opposite. When the patient's hand is resting, it is perfectly still. But when they initiate a voluntary movement, an intention to act, like reaching out to grab a glass of water, the tremor begins.
Why does it happen only when they move?
Because the cerebellum operates on a continuous feedback loop. As your hand moves toward the glass, the cerebellum is constantly calculating the distance and sending tiny corrective signals to keep the trajectory straight. A little to the left, a little to the right.
Wow.
When the cerebellar white matter is demyelinated, those corrective signals are delayed or lost. The brain overcompensates. The hand swings too far to the left, then overcorrects too far to the right.
The clinical term for that is dysmetria, right? Past pointing.
Exactly. As the hand gets closer and closer to the target, the need for precise correction increases, so the swinging gets wider, more erratic, and more violent. The tremor gets worse as it nears the glass. It is a profound manifestation of the loss of high-speed feedback.
Okay, we've mapped the clinical symptoms, but there is a phenomenon associated with MS that I find genuinely confusing, and I want to push back on the mechanics of it. I was reading about the Ethoff phenomenon. It's universally taught that a patient with MS shouldn't take hot showers or exercise in the heat because raising their body temperature can cause their neurological symptoms to suddenly return. Does heat actually trigger the immune system? Like, does taking a hot bath cause the T cells to suddenly launch a new attack and destroy more myelin?
It is a critical distinction to make, both for understanding the disease and for reassuring patients. No, the heat absolutely does not trigger a new immune attack. A hot shower does not destroy a single molecule of myelin. What you're describing is what we call a pseudo-relapse.
Pseudo-relapse. So if no new damage is occurring, why does the patient suddenly go blind again or lose the ability to walk?
We have to go right back to the fundamental physics of the axon we discussed in the first section. Think about that demyelinated nerve. The insulation is gone. The resistance is low. The current is leaking out.
The wire's already hanging by a thread.
Exactly. Even at a normal body temperature of 98.6 degrees, that nerve is barely managing to propagate the action potential. The safety factor, the amount of excess current available to guarantee the signal fires, is practically zero.
Wow.
Now, when you introduce physical heat, whether from a hot shower, a fever, or a rigorous workout, you physically alter the thermodynamics of the nerve membrane.
Wait, heat changes the physics of the cell?
It subtly alters the kinetics of the voltage-gated sodium channels. As temperature rises, these channels actually open and close slightly faster. In a normal, healthy, myelinated nerve, this means absolutely nothing. The safety factor is massive. There is more than enough current to trigger the next node, regardless of temperature.
Right, it's insulated.
But for a demyelinated nerve that is already starved for current, that tiny shift in channel kinetics is disastrous. The channels close too quickly before enough sodium can rush in to regenerate the signal.
Oh, the threshold isn't met. The tiny trickle of current is no longer enough to bridge the gap.
The heat pushes that damaged nerve over the edge into a state of total conduction failure. The wire completely shorts out, the signal stops entirely. And clinically, whatever old symptom that nerve was controlling, vision, motor strength, sensation, suddenly and dramatically returns.
But the beauty of it is it's entirely temporary.
Completely reversible. Once the patient steps out of the shower or drinks ice water and their core body temperature normalizes, the thermodynamics of the membrane stabilize. The sodium channel kinetics return to baseline.
Right.
The fragile conduction is restored, and the symptoms vanish just as quickly as they appeared. A true relapse, on the other hand, requires a new wave of inflammation. New macrophages destroying new myelin, resulting in neurological deficits that last longer than 24 hours in the absence of any fever or heat stress.
That is fascinating physics. The heat doesn't create the damage, it just exposes the broken wire that was already there. So this naturally leads to the next massive question. If symptoms can come and go, even in true relapses where myelin was actively destroyed, what is happening at the cellular level to fix these broken wires? How does a patient suddenly regain their vision a month after an attack? We need to look at the reversibility window. What is the repair mechanism?
This is where we shift from the pathology of destruction to the biology of repair. And this is a highly active area of research. As we established earlier, mature myelin itself does not spontaneously regenerate. If a macrophage digests a mature oligodendrocyte, that cell is dead and gone forever.
But patients with the most common form of the disease, relapsing remitting MS, they do recover. Their symptoms peak, and then over weeks or months, they get better. Sometimes they recover baseline function completely. If the workers are dead, who is replacing the insulation?
It turns out the adult central nervous system is not entirely devoid of regenerative capacity. Scattered throughout the white matter residing in a dormant state are populations of adult stem cell-like cells called oligodendrocyte precursor cells, or OPCs. You'll sometimes see them referred to in the literature as NG2 positive cells.
Precursor cells. So these are the reserves waiting on the bench.
Precisely. They're immature utility workers. When an acute inflammatory attack finally burns out and the T cells retreat, the local microenvironment changes. The damaged tissues release growth factors and chemical attractants.
Okay.
The OPCs sense this distress signal. They break out of their dormant state, physically migrate through the brain tissue directly into the center of the demyelinated plaque, and they begin to rapidly divide and proliferate.
They mobilize a massive temp workforce, and then they differentiate.
Yes, they mature into brand new, fully functional oligodendrocytes. They extend their arms, find the naked exposed axons, and they begin to lay down fresh layers of lipid membrane. They remyelinate the wire.
And as they wrap the wire, the membrane resistance goes back up, the capacitance goes back down, and saltatory conduction is restored. The patient regains the ability to walk or see. But there's a catch here, isn't there? Because the remyelination process isn't perfect. I read an analogy. If the original myelin laid down during your childhood development is this thick, heavy-duty, perfectly engineered industrial rubber cable, the repair job done by the OPCs is like wrapping the wire in cheap, thin electrical tape. It works, but it's structurally inferior.
That analogy captures the histology perfectly. When a pathologist looks at a remyelinated plaque under an electron microscope, we call it a shadow plaque, it looks distinctly different. The OPCs do their best, but the remyelination is suboptimal.
In what way?
The new myelin sheaths are physically much thinner than the original ones. We measure this using something called the G ratio, which is the ratio of the inner axonal diameter to the total outer diameter. In repaired myelin, the G ratio is abnormally high, meaning the myelin is too thin.
So the insulation isn't as robust. What else is wrong with it?
The internodes, the length of myelin between each bare node of Ranvier, are significantly shorter.
Meaning the electrical signal has to stop and actively regenerate at the booster stations much more frequently.
Exactly. So while conduction is successfully restored, it is permanently less efficient. The safety factor is permanently lowered. The nerve fires slower and requires more energy.
That makes sense.
This explains why recovery from a relapse is frequently incomplete. The patient might regain their vision, but colors might appear slightly washed out forever, or they might have a lingering subtle weakness in their leg. The system has been patched, but it is fundamentally altered.
And this mechanical reality brings us to the core tragedy of the disease progression. Early on, in the relapsing remitting phase, this reversibility window is open. The myelin is stripped away, but the physical copper wire, the axon itself, survives the attack. So the OPC temp workers can rush in and apply the electrical tape. But eventually, in almost all patients, the disease shifts. The remyelination fails, the deficits become permanent, and the patient steadily declines. What exactly pushes the pathology from reversible myelin damage to irreversible brain damage?
The burden of chronic inflammation and energy failure. Early on, the axon is incredibly resilient. It can survive being naked for a while, but repeated inflammatory attacks subject the axon to intense, persistent oxidative stress. The macrophages are constantly dumping reactive oxygen species and nitric oxide onto the bare wire.
It's basically bathing in toxic exhaust.
Furthermore, remember that an unmyelinated nerve is terribly inefficient. To keep the signal moving without insulation, the axon has to drastically upregulate the number of sodium-potassium pumps on its membrane. These pumps require massive amounts of ATP cellular energy to function. The mitochondria inside the axon are working in overdrive to produce this energy.
But mitochondria need oxygen, and an inflammatory plaque is a highly hypoxic, suffocating environment.
Precisely. The mitochondria eventually fail. ATP production plummets. When ATP is depleted, the sodium-potassium pumps fail. Sodium builds up massively inside the axon.
Oh, no.
Now, the cell has an emergency backup system, the sodium-calcium exchanger, which normally pumps calcium out of the cell. But because the sodium concentration is so disastrously high inside, the exchanger runs in reverse. It pumps sodium out, but it pumps massive toxic amounts of calcium into the delicate interior of the axon.
Calcium is usually tightly regulated. A huge influx of calcium is a universal death signal for a cell.
He is the executioner. The flood of calcium hyperactivates a class of destructive enzymes called calpains. Calpains are essentially molecular scissors. They start aggressively cleaving and digesting the neurofilaments and microtubules that make up the physical internal skeleton of the axon. The structural integrity of the wire collapses. Eventually, the axon undergoes complete transection. It snaps in half.
The wire is cut.
And once the axon is severed, the distal portion undergoes Wallerian-like degeneration. It disintegrates. Right. And here's the brutal truth. Once the axon is gone, there is absolutely nothing left to remyelinate.
The OPCs have no substrate to wrap.
Right. The electrical tape cannot fix a severed wire. The neurological deficit is now permanently locked in.
And what happens to that microscopic graveyard of dead axons? Does the brain just leave an empty void there?
The central nervous system aggressively attempts to wall off the damage to protect the surrounding healthy tissue. It utilizes its primary support cells, the astrocytes. Normally, astrocytes are peaceful cells that regulate synapses and maintain the blood-brain barrier. But in the presence of severe neuronal death, they undergo massive hypertrophy. They proliferate, enlarge, and upregulate a structural protein called glial fibrillary acidic protein, or GFAP.
So they transform from peaceful support cells into a biological rapid response concrete crew.
They lay down a dense, tangled, fibrotic-like web over the entire area. This process is called astrocytic gliosis. They literally pave over the ruins of the dead axons.
They form a hard scar, a sclerosis.
Yes, that is exactly where the sclerosis in multiple sclerosis comes from. When a pathologist touches the brain during an autopsy, or a radiologist looks at a late-stage MRI, they see and feel these multiple hard, firm, gliotic scars littered throughout the white matter.
And at this point, the clinical diagnosis shifts.
It transitions from relapsing remitting MS to secondary progressive MS. The acute inflammatory fires might actually quiet down, you see fewer relapses, but the patient's neurological decline continues relentlessly, a slow, steady downward slope as the functional reserve of the brain is finally exhausted and the severed axons cannot be replaced.
It's a sobering mechanical reality. The duct tape can only hold the overworked wire together for so long before the wire itself rots away from energy starvation. We have dug deeply into the physics, the immunology, and the cellular destruction. But now we have to pivot and ask, who is this happening to? Let's look at the epidemiology and the evolutionary biology of MS. Because if you read a board exam question, the classic clinical vignette is almost always identical: a young woman, usually between the ages of 20 and 40, often of Northern European descent.
The epidemiology is striking and highly specific. Multiple sclerosis is a disease that overwhelmingly affects women. The incidence is roughly 3 times higher in women than in men. And for centuries, the medical community's interpretation of this data was, frankly, backwards and rooted in deep misogyny.
Here's where it gets really interesting. I was reading the historical context, and in the 19th and early 20th centuries, physicians looked at autoimmune diseases predominantly affecting women and concluded that women simply had weaker nerves, a fragile constitution, or a biological predisposition to hysteria. But immunologically speaking, the reality is the exact opposite of weakness, right?
It is the absolute opposite.
Yes.
Women do not get multiple sclerosis because their systems are weak or fragile. They get it because they possess an incredibly powerful, hypervigilant, over-optimized immune system.
Right.
If we return to our military analogy, the female immune system is not a small, disorganized militia. It is a massive, elite, highly alert, standing army that is primed to respond aggressively and rapidly to any perceived threat.
And why did evolution build the female immune system to be so much more aggressive than the male immune system?
It comes down to the brutal mathematics of human survival. Evolutionarily, women carry an immense biological burden. They must survive infectious diseases not just for themselves, but to protect the fetus during pregnancy.
Wow.
Furthermore, mammalian survival requires the mother to pass on a highly diverse, robust repertoire of antibodies through breast milk to keep the newborn infant alive until its own rudimentary immune system can develop.
So women biologically needed a supercharged defense force to ensure the survival of the species.
Exactly. And the data bears this out. Women, on average, mount significantly stronger CD4-positive T-cell responses than men. Their B cells are more active, and they produce substantially higher baseline levels of circulating antibodies.
That's incredible.
They respond better to vaccines, generating higher protective titers, and they generally clear acute viral and bacterial infections faster and more efficiently than men.
But biology is always a trade-off. There is no free lunch in evolution. The trade-off for having an elite, hair-trigger military constantly patrolling the city is a dramatically higher risk of friendly fire.
Precisely. Because the female immune system is so highly reactive, so primed to attack, it is simply, statistically and biologically, more prone to losing self-tolerance. It is more likely to misidentify a benign, self-manufactured tissue, like the myelin basic protein on an oligodendrocyte, as a dangerous foreign invader and launch a devastating, unstoppable autoimmune attack.
And the generals commanding this elite army are heavily influenced by hormones, right? Let's talk about the role of estrogen, because this is where the endocrinology intersects with the immunology in a way that is just wild. Estrogen doesn't just have one simple effect. Its impact on the immune system completely depends on the concentration.
It's a brilliant, highly dynamic system. At normal baseline menstrual cycle levels, estrogen generally acts as an immune enhancer. It supports that adaptive immune activation, it promotes the survival of B cells, enhances antigen presentation, and maintains the baseline susceptibility for autoimmunity. It keeps the army on high alert.
But then a woman gets pregnant. And over the course of gestation, estrogen and progesterone levels absolutely skyrocket to concentrations thousands of times higher than baseline. What happens to the elite army then?
The immune system faces a massive biological paradox. A fetus is technically an allograft, a genetically foreign entity growing inside the body. Half of the fetus's DNA comes from the father, meaning the fetus is expressing foreign proteins.
Right.
If the mother's elite, hyperaggressive immune system stayed fully active, it would immediately recognize the fetus as a parasite, attack it, and reject it.
It would destroy the pregnancy. So how does the body prevent that?
The skyrocketing levels of estrogen and progesterone during pregnancy bind to receptors on the immune cells and force a massive systemic shift in the immune profile. The hormones issue a direct, overriding stand-down order to the troops.
Wow.
They suppress the aggressive inflammatory Th1, Th17 cells, the exact cells that drive MSAC. Simultaneously, they massively upregulate the production of regulatory T cells, or TREGs, and shift the balance toward a Th2 anti-inflammatory tolerant state.
The military is forced to pack up their weapons and go back to the barracks to keep the peace for the baby.
Exactly. And the clinical result of this profound immune suppression is miraculous for MS patients. Women with MS almost universally experience a dramatic, profound decrease in the rate of clinical relapses during pregnancy, particularly in the third trimester when hormone levels peak. The disease practically goes into total remission.
The patients actually feel better. The friendly fire stops completely, but pregnancy ends. The baby is born.
Delivery occurs. The placenta is expelled. And within a matter of days, the massive suppressive tidal wave of estrogen and progesterone crashes violently back down to baseline levels.
The stand-down order is abruptly revoked.
And the immune system rebounds with a vengeance. It wakes up, realizes it has been artificially suppressed for 9 months, and the inflammatory Th1 and Th17 cells hyperactivate.
Oh, wow.
This postpartum period, specifically the first 3 to 6 months after birth, is statistically the most dangerous time for a woman with MS. It is associated with a drastically increased risk of a severe, sometimes debilitating clinical relapse.
It is the ultimate slingshot effect. You pull the aggressive immune system back against its will for 9 months, and the moment you let go of the tension, it snaps forward with terrifying momentum and obliterates the myelin. That pregnancy postpartum cycle is the clearest possible indicator of how deeply hormonally driven this autoimmune dynamic is. But it's not just the fluctuating hormones, is it? The immune aggression is literally woven into the structural genetics of being female.
Yes, the fundamental chromosomal architecture, XX versus XY. The X chromosome is not just about sex determination, it is incredibly rich in immune-regulating genes. It contains genes governing toll-like receptors, FOXP3 for regulatory T cells, and various cytokine pathways. Now, in biological females who possess 2 X chromosomes, the body employs a process called X inactivation. Early in embryonic development, 1 X chromosome in every cell is randomly crumpled up and silenced into a bar body to ensure females don't get a toxic double dose of gene products compared to males.
Right, you only need one set of instructions active at a time.
However, the X inactivation mechanism is imperfect. About 15% of the genes on that silenced X chromosome manage to escape inactivation. They stay active.
Ah,
And crucially, many of those escapee genes are the immune regulators, like TLR7. Because women have two X chromosomes, they end up with a structurally higher baseline expression of these specific immune-stimulating genes compared to men who only have one X chromosome. It is a hardwired, permanent genetic push toward higher immune reactivity.
so women have the genetics and the hormones pushing them toward the disease, but men do get MS. It's much less frequent, roughly a 3-to-1 ratio, but men absolutely do develop it. And there is a clinical paradox surrounding male patients, isn't there?
The male paradox of MSS. Men are statistically protected from initiating the disease. This is likely due to their lack of the second X chromosome, their lower baseline immune reactivity, and the constant presence of testosterone, which is generally a highly immunosuppressive hormone. It keeps the immune system relatively quiet.
Right.
But if a man's immune system does manage to overcome those significant barriers and trigger the autoimmune cascade, the resulting disease course is often much more severe.
Why is it worse if it's harder to start?
We are still unraveling the precise molecular mechanisms, but the clinical data is clear. Men are significantly more likely to present with primary progressive MSS day, meaning they skip the relapsing remitting phase entirely and just experience a steady, relentless decline from day one.
That's awful.
They accumulate physical disability faster, they suffer greater irreversible axonal transection earlier in the disease course, and their long-term prognosis for maintaining mobility is generally poorer.
So it's much harder to start the fire in a man's central nervous system, but once the fire is finally burning, it is incredibly difficult to put out. It burns hotter and destroys the infrastructure faster. Perhaps they lack the regenerative OPC capacity, or perhaps without the estrogen fluctuations, the immune system never gets a break. Okay, so we have built a comprehensive profile of the vulnerable patient, genetically susceptible, possessing a robust, hypervigilant immune system. But what actually pushes them over the edge? People aren't born with active MS, it develops later in life. What is the spark that ignites the fire? This brings us to our final exploration: triggers, diagnosis, and management.
MS requires a perfect storm of factors colliding. It begins with an underlying genetic susceptibility. While MS isn't purely hereditary in a Mendelian sense, genetics heavily load the gun. The most famous and heavily validated genetic link is the HLA-DRB 115.01 allele.
HLA, human leukocyte antigen. This is part of the major histocompatibility complex, or MHC class II. These are the protein hands that immune cells use to present captured evidence to the T cells.
Exactly. The MHC class II molecule has a peptide-binding cleft. Its job is to grab a piece of a digested protein and hold it up for the T-cell receptor to inspect. If you inherit the HLA-DRB 115.01 variant, the specific physical shape of your peptide-binding cleft happens to be perfectly contoured to present myelin basic protein in a way that highly activates those autoreactive T cells. The genetic architecture makes the mistake more physically probable.
But genetics alone are not destiny. Having the gene doesn't mean you will absolutely get the disease. You need an environmental trigger to pull the trigger on the loaded gun. And looking through the recent blockbuster research, there is a specific virus that keeps showing up at the scene of the crime: Epstein-Barr virus, or EBV.
The Epstein-Barr virus connection is arguably the most exciting, paradigm-shifting area of MS research in the last decade. Now, EBV is incredibly common. It's the virus that causes infectious mononucleosis, and over 90% of the global human population is infected with it at some point in their lives. The virus goes dormant and hides forever inside your B cells.
If everyone has the virus, how can it cause a rare disease like MS?
Because in that small subset of genetically susceptible people, the immune system's initial battle against the EBV virus goes terribly, tragically wrong through a biological phenomenon known as molecular mimicry.
Molecular mimicry, meaning the virus is wearing a biochemical disguise that looks identical to the body's own tissue.
Yes. A few years ago, researchers conducted a massive longitudinal study tracking millions of military personnel. They definitively proved that EBV infection precedes the onset of MSAria. Then they looked at the molecular structure. It turns out that a specific protein manufactured by the Epstein-Barr virus called EBNA1 shares an almost identical structural homology, the exact same sequence of amino acids with a protein found in the central nervous system called GLIAL-CAM, and it's also highly similar to myelin basic protein.
They share the exact same protein barcode.
Precisely. When the patient gets mono, their immune system generates massive amounts of high-affinity antibodies and highly trained T cells designed specifically to hunt down and destroy the EBNA1 protein to clear the virus. The military builds a hyper-specific weapon.
But because of the molecular mimicry, the weapon is cross-reactive.
The military gets confused. They successfully suppress the virus. But those highly trained memory T cells and antibodies continue to circulate. Eventually, they cross the blood-brain barrier, encounter the myelin basic protein, and the T cell receptor binds to it. The T cell thinks, the Epstein-Barr virus is back. Attack. It unleashes the full destructive cascade we discussed earlier, completely unaware that it's actually destroying the patient's own neural insulation because the myelin is wearing the same uniform as the virus.
That is deeply tragic biology. The immune system is just trying to protect the host from a virus, and it accidentally triggers a lifetime of demyelination. And there are other environmental factors that fuel this fire, right? Vitamin D and smoking.
Yes, vitamin D is a potent immune modulator and naturally suppresses inflammatory Th1 cells and boosts regulatory T cells. The promoter region of the HLA gene we just discussed actually has a vitamin D response element. This perfectly explains the geographic epidemiology of MS. The incidence drastically increases the further you live from the equator, where sunlight exposure and subsequent natural vitamin D synthesis are much lower. And smoking simply creates a massive, chronic, highly pro-inflammatory state in the lungs, activating peripheral immune cells and constantly keeping the army on an agitated hair trigger alert.
So we have the perfect storm, the genetics, the viral mimicry, the low vitamin D. The storm hits, the T cells breach the barrier, and a young woman comes into the neurology clinic with sudden, painful blindness in one eye optic neuritis. Or she has the violent nystagmus of an internuclear ophthalmoplegia. The clinician suspects mammoth. How do they lock in the diagnosis? You mentioned the McDonald criteria earlier, which demands proof of dissemination in time and space.
Clinical symptoms are the starting point, but modern diagnosis relies overwhelmingly on advanced neuroimaging, specifically magnetic resonance imaging, or MRI, of the brain and spinal cord, utilizing a specialized intravenous contrast agent called gadolinium.
Let's break down the physics of the MRI contrast, because it directly ties back to the pathology we discussed. Why gadolinium? What does the dye actually do?
Gadolinium is a heavy metal with paramagnetic properties that make it shine brightly on a T1-weighted MRI sequence. But crucially, gadolinium is a large molecule. If you inject it into the bloodstream of a healthy person, it circulates through the brain's blood vessels, but it absolutely cannot cross the intact, tight junctions of the blood-brain barrier. It stays inside the pipes.
But remember our rogue T cells? Remember the matrix metalloproteinases? They used molecular blowtorches to melt the tight junctions and blast the doors open to get into the brain.
Exactly. An active, acute MS plaque is characterized by a violently disrupted, leaky blood-brain barrier. So when you inject the gadolinium, it circulates to that exact spot, leaks out of the damaged vessel into the brain tissue, and gets trapped in the active plaque. When you run the MRI, that specific active lesion will glow brilliantly bright white.
It highlights the active fire. But what about an old plaque? A demyelinated scar from an attack that happened 3 years ago?
In an old, burned-out plaque, the myelin is gone and the axon might be dead, but the acute inflammation is stopped. The astrocytes have moved in, formed the glial scar, and successfully repaired the blood-brain barrier. The doors are sealed again.
Right.
So when the gadolinium flows past an old plaque, it cannot leak out. The old plaque will show up as a dark shadow, a black hole on a T1 scan, but it will not enhance with contrast.
That is diagnostic gold. Because if you do a single MRI scan on a patient today and you see 5 lesions scattered across the brain, meaning you have dissemination in space, and 2 of those lesions are glowing brightly with gadolinium, while the other 3 are dark, non-enhancing holes, you have instantly proven dissemination in time on a single scan.
You have incontrovertible visual proof of old scars and new fires occurring at the exact same moment. Combine that MRI finding with lumbar puncture showing the elevated IgG index and the oligoclonal bands we discussed, and your diagnosis of multiple sclerosis is formally locked in.
So the diagnosis is made. The reality sets in. How do we fight back? What is the management strategy? Because as of today, we cannot magically regrow severed axons, and we cannot entirely cure the disease.
Management is clinically divided into 2 distinct, non-overlapping phases, putting out the immediate acute fire and fireproofing the forest to prevent future fires. If a patient comes into the emergency room having an acute relapse, they suddenly can't walk, or they have lost their vision, we must act immediately to stop the active demyelination and save the axon from dying.
And what is the weapon of choice for an acute fire?
We use massive, high-dose intravenous corticosteroids, typically methylprednisolone, administered over 3 to 5 days.
We are talking about carpet bombing the immune system. How do steroids actually stop the attack on a cellular level?
Corticosteroids are incredibly potent, broad-spectrum immunosuppressants. They physically cross the cell membrane of the rogue T cells and B cells, bind to the glucocorticoid receptors in the cytoplasm, and translocate directly into the nucleus. Once in the nucleus, they alter the transcription of the cell's DNA. They massively downregulate the production of inflammatory cytokines like interferon gamma and TNF-alpha. They upregulate anti-inflammatory proteins.
They silence the communication network.
Furthermore, they actively close the blood-brain barrier, preventing new troops from entering. And most importantly, they force the hyperactive lymphocytes currently inside the brain to undergo apoptosis. They literally trigger a genetic suicide pathway within the rogue cells. The active inflammation halts, the swelling goes down, and the physiological environment is stabilized so the OPCs can begin their remyelination work.
The fire is out. But you cannot leave a human being on massive suppressive doses of IV steroids forever.
Absolutely not. The side effects, bone necrosis, massive weight gain, diabetes, overwhelming susceptibility to fatal infections would be catastrophic. Steroids are for acute rescue only. For the long-term management of the disease, we transition the patient to disease-modifying therapies, or DMTs.
And the landscape of DMTs has completely revolutionized MS care. We have moved so far beyond the older, mildly effective interferon injections that just broadly dampen the immune system with flu-like side effects. Today, we use highly targeted monoclonal antibodies biologics. We are basically sending in biological snipers to take out specific members of the rogue union. Let's look at natalizumab.
Natalizumab is a brilliant piece of rational drug design. Remember the breach. The T cells use that specific Velcro molecule, alpha-4 integrin, to stick to the blood vessel walls and cross the blood-brain barrier. Natalizumab is an antibody engineered in a lab to specifically bind to and cover up the alpha-4 integrin on the surface of the T cells.
It covers the Velcro with smooth tape.
Exactly. The rogue commanders are still floating around in the bloodstream, highly aggressive and fully armed, but when they try to stick to the brain's blood vessels, they slide right off. They are physically locked out of the central nervous system. It is incredibly effective at stopping relapses.
Though it does come with a terrifying risk of PML, progressive multifocal leukoencephalopathy, because by completely locking the immune system out of the brain, you allow dormant viruses, like the JC virus, to wake up and destroy the brain unchallenged, which just highlights how dangerous it is to mess with immune traffic. What about targeting the B cells? We spent a long time proving that B cells are the localized antibody factories throwing the inflammatory party.
This is where drugs like ocrelizumab have changed the world. Ocrelizumab is a monoclonal antibody that specifically targets a protein called CD20, which is found exclusively on the surface of mature B cells. When the drug binds to the CD20 receptor, it marks that B cell for death. The body's own natural killer cells come in and destroy it via antibody-dependent cellular cytotoxicity.
You are systemically assassinating the B cell population.
We wipe them out. And by depleting the B cells, we shut down the antigen presentation to the T cells. We shut down the intrathecal antibody production, and we silence the inflammatory signaling. Drugs like ocrelizumab have been revolutionary because they are not only highly effective at reducing relapses and relapsing remitting MS, but they were the very first drugs proven to significantly slow down the insidious decline of primary progressive MS.
They don't cure the disease. They can't bring back the dead axons or dissolve the hard astrocytic scars. But they drastically reduce the frequency of the fires, preserve the physical infrastructure for decades longer, and completely alter the trajectory of a patient's life. It is an incredible triumph of modern medical science, built entirely on understanding the microscopic mechanics we've explored today.
It is a testament to what we can achieve when we stop looking at a disease as a generic failure.
Progressive multifocal leukoencephalopathy, or PML.
PML.
We're starting with a scenario where the house's security system is completely disabled. The front door is blown off its hinges, the guards have all gone home, and the property is left wide open.
Left wide open for an opportunistic assassin, one that was actually hiding in the basement the entire time.
And that assassin is the JC virus, right?
Yeah.
Named after John Cunningham, the first patient it was isolated from.
Yeah. Now, when people hear the phrase demyelinating disease of the brain, the immediate assumption is multiple sclerosis.
Right. MS is super famous.
It is the most famous disease in this category. But the clinical texts draw a massive, heavy line between MS and PML.
Because they operate so differently.
Entirely differently. Multiple sclerosis is an autoimmune condition. The body's own immune system gets confused and mistakenly attacks the myelin.
But PML is different.
Very. Progressive multifocal leukoencephalopathy is not autoimmune in any way. It is a progressive, relentless viral infection.
So it's the virus doing the damage directly.
Like the JC virus directly infects the oligodendrocytes, replicates millions of copies of itself inside them, and then lyses or literally explodes the cells to spread further.
Jeez. So in MS, the immune system is accidentally stripping the rubber insulation off the wires. But in PML, this virus is walking room to room, directly murdering the electricians.
That is exactly it, and it does so with incredible stealth.
Which is wild, because it's not some exotic bug, right?
Not at all. It's not some rare insect bite. The source materials highlight a shocking statistic. 50 to 80% of adults walking around right now, most of the people listening to this deep dive, are already infected with the JC virus.
Wait, really? Most of us have it?
Yes. It's a very simple, circular, double-stranded DNA virus. Most of us pick it up during childhood, usually without a single symptom.
And then what? It just hangs out?
Basically.
Yeah.
The immune system handles the initial exposure, and then the virus simply goes into hiding. It enters a state of deep latency.
Where does it hide?
Just sitting completely dormant inside the tissues of your kidneys, your bone marrow, and your lymphoid organs.
Okay, wait, hold on. If 80% of us have this JC virus just hanging out in our kidneys right now, why aren't healthy people constantly dropping from this?
It's a great question.
I mean, viruses mutate constantly to survive, right? Why hasn't this thing figured out how to bypass a healthy immune system and attack the brain?
Because the JC virus is a pure opportunist. It lacks the sophisticated immune evasion machinery that you see in something like, say, HIV.
Yeah.
It is completely and utterly vulnerable to your cellular immune system, specifically your CD8 killer T cells.
Our internal guards.
Right. Think of those T cells as heavily armed wardens patrolling the kidneys and bone marrow. The second a dormant JC virus tries to wake up and produce a viral protein, a patrolling T cell recognizes it.
And just takes it out.
Immediately. It destroys that host cell before the virus can even replicate. The virus cannot win a fight against a healthy immune system.
It can only attack when the opponent isn't even in the ring.
Precisely.
Which totally explains why the texts are so adamant that PML almost exclusively occurs in severely immunosuppressed patients.
Yes, exactly.
Like the classic textbook scenario is an HIV's patient whose disease has progressed so far that their CD4 T cell count drops below 200 or even below 100.
The wardens are essentially wiped out by the HIV.
The cellular immunity just completely collapses. But there is another highly tested clinical scenario in modern medicine that triggers PML. And this is wild. It involves patients who actually have multiple sclerosis.
Right, the natalizumab patients.
Yeah. So doctors treat severe MS with this highly effective drug called natalizumab. It's a monoclonal antibody that targets a protein called alpha-4 integrin.
Which sounds super complicated.
But to put it simply, alpha-4 integrin is the molecular key that allows immune T cells to open the door of the blood-brain barrier.
Right, lets them cross from the bloodstream into the brain tissue.
Right. So in MS, you want to block those T cells from getting into the brain because they are the ones causing the autoimmune damage to the myelin.
Okay, so the drug confiscates their keys.
Exactly. The T cells are locked out of the brain, the autoimmune riot stops, and the MS patient feels better.
They feel much better. But by completely locking the immune system out of the central nervous system, you have inadvertently created a biological safe haven. Yeah. If the JC virus wakes up in the kidneys, perhaps because of a minor dip in overall immunity, it enters the bloodstream.
And when it reaches the blood-brain barrier,
It finds no T cells guarding the gates. It slips inside. And once it is inside the brain tissue, there is absolutely zero immune surveillance to stop it.
that is a staggering medical irony. You lock the security guards outside the house to stop them from accidentally breaking the furniture.
Right.
But in doing so, you leave the vault totally unguarded against the silent thief who just climbed in through a window.
And once the JC virus is inside the central nervous system, its behavior is ruthlessly efficient.
It just goes to work.
It specifically targets the oligodendrocytes. It binds to their surface receptors, gets engulfed into the cell, and transports its viral DNA straight into the nucleus.
Basically hijacking them.
Completely. Instead of making myelin, the cell is forced to replicate millions of viral DNA copies and build new viral protein shells.
Ugh, that's brutal.
It is. The nucleus actually swells up with these viral inclusions. It grows massive and distorted until the cell membrane simply cannot hold it anymore.
It pops.
The oligodendrocyte ruptures, dies, and releases a swarm of new viruses to infect the neighboring electricians.
And when the electrician dies, the myelin insulation they were maintaining disintegrates.
Exactly.
The electrical current leaks out of the raw copper axons, and conduction completely fails. And the name of the disease gives away exactly how this looks in the patient.
Progressive, multifocal leukoencephalopathy.
Let's break that down. Progressive means it relentlessly worsens, right? There are no periods of recovery.
Correct.
Leuko means white matter. Encephalopathy means brain dysfunction. But it's the word multifocal that is so devastating here.
Because the virus crosses the blood-brain barrier in various random locations, it establishes multiple beachheads.
So it attacks completely independent areas of the brain's white matter all at the same time.
And the clinical presentation is incredibly chaotic because of that multifocal nature. A patient won't just have one specific deficit.
It's all over the place.
The symptoms depend entirely on which rooms of the house are currently losing power.
Wow.
If the virus is lysing oligodendrocytes in the occipital lobe at the back of the brain, the electrical signals from the eyes can't be processed.
So they go blind.
They develop visual field defects or even total cortical blindness, despite their actual eyes working perfectly fine.
Because the wire connecting the camera to the computer is stripped.
Exactly. Meanwhile, if another viral colony is actively destroying the frontal lobe, you see profound cognitive decline, sudden personality changes, a loss of executive function.
So scary.
If it hits the parietal lobes, the patient experiences bizarre sensory deficits, like an inability to process touch or recognize objects by feel.
All at the same time.
Yes. And if it infiltrates the descending corticospinal tracts, the main motor highways, you see classic upper motor neuron signs, extreme weakness, spasticity, and hemiparesis, which is paralysis on one side of the body.
And all of this is happening to the patient simultaneously, just continually getting worse week by week.
It's a terrifying clinical picture.
It really is. The USMLE prep materials actually provide a really dark but highly effective mnemonic to memorize all this.
Oh, the JC kills one.
Yeah. JC kills oligodendrocytes.
That's very memorable.
J stands for JC virus. C stands for cellular immunity failure. K stands for kills the cells. O stands for opportunistic infection. L stands for leukoencephalopathy, and so on.
It basically maps out the entire pathology.
But I want to talk about how a doctor actually confirms this is happening inside a living patient's skull. Let's talk about the MRI scans.
The radiologic findings in PML are a perfect, haunting reflection of the underlying biology.
What do they look like?
When you look at an MRI of a healthy brain, the white matter looks uniform. But radiologists use specific imaging sequences like T2 and FLAIR.
Which are like filters, right?
Yeah, think of T2 and FLAIR as biological filters on the camera that are specifically designed to make water and inflammation glow bright white on the screen.
Okay, got it.
When a radiologist looks at a PML patient's brain using these filters, they see multiple asymmetric patches of glowing white matter.
So just random spots.
Right. These lesions don't follow any specific blood vessel territory. They just look like random spreading clouds of damage.
But the sources point out two massive, glaring negative findings on the MRI that are the real key to the diagnosis. They say there is no mass effect and no ring enhancement.
This is where the biology gets really fascinating.
Let's unpack that jargon because I think it's so interesting. Usually, if you have a massive infection in your brain like a bacterial abscess, your immune system rushes in to fight it.
Naturally.
That causes a ton of swelling, right? Pushing on the surrounding brain tissue. That pushing is called mass effect.
Exactly.
And if the doctor injects a contrast dye into your veins for the MRI, the intense inflammation causes the blood vessels to leak the dye, creating a bright white ring around the infection. That's ring enhancement.
But in PML, neither of those things happen.
Which is wild.
There is no mass effect pushing the brain structures out of the way. And there is absolutely no ring enhancement when they inject the contrast dye.
Because there is no immune system left to cause the inflammation.
Yeah, exactly. You are witnessing active cellular destruction in a biological vacuum.
A vacuum? Wow.
The JC virus is slaughtering the oligodendrocytes by the millions, and the body isn't even mounting a response.
The fire alarms are completely silent.
There's no swelling, no edema, no inflammatory capsule trying to contain the spread, just quiet, progressive demyelination.
And if you were to look at that tissue under a microscope, pathologists describe this very specific, chaotic scene.
Oh, it's a mess in there.
They see the naked, uninsulated axons. They see those enlarged, swollen oligodendrocyte nuclei packed with viral particles.
And the foamy macrophages.
Yeah. What are those?
They're basically cellular garbage trucks that have arrived to quietly swallow up the fatty myelin debris. They get so bloated with fat that they literally look like seafoam under the scope.
That's disgusting but fascinating. And they also see bizarre reactive astrocytes.
Right. Astrocytes are the structural support cells of the brain.
And in PML, they get so stressed and deformed by the surrounding destruction that they mutate into these giant multinucleated shapes, right?
Yeah, they get so deformed they can actually trick a doctor into thinking they're looking at a highly aggressive brain cancer like glioblastoma.
It is a microscopic war zone where one side didn't even show up to fight.
And because it's a virus, antibiotics do absolutely nothing.
Right. The medical texts state that there is no specific antiviral drug that cures PML.
Sadly, no.
The only treatment strategy is to somehow desperately turn the patient's immune system back on.
If they have HIV, you blast them with highly active antiretroviral therapy to get their CD4 count back up.
And if they're the MS patient on natalizumab,
You stop the drug immediately. And you might even use plasma exchange to filter the remaining drug out of their blood just to let the T cells back into the brain.
so it basically becomes a race against time. You are praying the immune system reboots fast enough to cross the blood-brain barrier and clear out the JC virus before the naked axons begin to snap.
Because while oligodendrocytes can slowly remyelinate a bare wire,
If the raw copper wire itself degenerates and dies from a lack of support, that neurological deficit is permanent.
once the axon dies, it's over.
So the JC virus exploits a completely unguarded nervous system, but nature loves a cruel irony, right?
Definitely.
Because it turns out having a fully armed, hypervigilant immune system can actually be just as destructive to your myelin.
Sometimes even more so.
In fact, the very weapons your body builds to save your life can turn around and burn your own house down. So let's pivot from PML to our second disease, acute disseminated encephalomyelitis, or ADEM.
This is a massive shift in pathology.
Yeah, it really is.
While PML is a slow, progressive infection targeting the immunocompromised, ADEM is an explosive, acute, autoimmune riot. And it typically happens in patients who have completely healthy, robust immune systems. In fact, it classically occurs in young children.
Wait, healthy kids.
Yeah. Usually 1 to 4 weeks after they have successfully fought off a routine viral infection, like the measles or chickenpox. Or, less commonly, a few weeks after receiving a vaccination.
Okay, so a child catches a virus, gets better, and then weeks later, their brain's insulation is destroyed.
It sounds impossible.
The mechanism behind this is something the texts call molecular mimicry. And I want to spend some time here because it is one of the most fascinating and tragic concepts in immunology.
Let's walk through the timeline.
Yeah, so a child is exposed to the measles virus. The virus enters their body. The immune system detects it and immediately spins up its manufacturing lines.
It acts like a highly advanced weapons factory.
Right. It analyzes the specific proteins on the surface of the measles virus, the viral antigens, and it custom builds millions of T cells and B cell antibodies specifically designed to lock onto that exact protein shape and destroy it.
The immune system is performing flawlessly here. It mounts a massive defense, hunts down the measles virus, clears the infection, and the child's fever breaks. They recover.
But the immune system is cautious. It keeps a massive army of those custom-built measles-hunting T cells on high alert, patrolling the bloodstream just in case the virus tries to return.
But then the cruel biological coincidence happens.
The molecular mimicry.
Yes. It turns out that a normal, healthy protein sitting right on the surface of the myelin sheath in the central nervous system shares an incredibly similar amino acid sequence to the measles virus protein.
On a molecular level, they look almost identical.
Right. So the immune system's T cells circulating in the blood manage to cross the blood-brain barrier as part of their routine patrol. They bump into an oligodendrocyte, they scan the myelin protein, and because the structural shape is so similar to the measles antigen, they were trained to hunt.
The T cell receptors lock on.
Exactly. They lock on, and the T cells send out chemical alarm signals, cytokines, screaming to the rest of the immune system, "The virus is hiding in the brain! Attack!"
It is a catastrophic case of mistaken identity. People sometimes describe this as a biological SWAT team being handed a blurry photo of a suspect.
I've heard that analogy.
But I feel like that implies the immune system is incompetent. It's not a blurry photo. It's more like the innocent bystander, the myelin, is walking down the street wearing the exact same highly specific custom-made uniform as the bank robber.
That's a much better way to put it. The SWAT team is doing exactly what it was trained to do.
The immune system recruits massive amounts of inflammation, kicks down the doors, and starts slaughtering the innocent myelin cells, fully believing it is saving the child from a measles relapse.
And because the immune system is so powerful, This is an acute attack.
It happens fast.
Very fast. Remember, PML took weeks or months to progress. ADEM hits over a matter of hours or a few days.
Wow.
The T cells trigger widespread perivenous demyelination. This means the inflammation and myelin destruction explode outward from all the tiny veins inside the brain and spinal cord, wherever the white blood cells are exiting the bloodstream.
But let me pause you there, because anyone listening who knows someone with multiple sclerosis is probably thinking, wait a minute, MS is also caused by an autoimmune attack on the myelin. The immune system destroys the insulation.
True.
So if a doctor is standing in the emergency room looking at a child experiencing a sudden, massive demyelinating attack, how do they know they aren't just witnessing the very first flare-up of a lifelong case of multiple sclerosis?
This is a critical differential diagnosis, heavily emphasized in the medical boards. You have to be able to tell them apart.
So what's the difference?
There are two major differentiators. The first is time. Multiple sclerosis is defined as being disseminated in time and space. It has a relapsing remitting course over a patient's entire lifetime.
It keeps coming back.
Exactly. ADEM, however, is almost always monophasic.
Meaning one phase.
Right. It is a single, massive, one-time inflammatory riot triggered by that specific viral mimicry event. Once the immune system finally calms down, it rarely ever happens again.
Okay, so MS comes back, ADEM is usually one and done. But the doctor in the ER doesn't have a time machine to see the future. What is the immediate clinical difference right there in the room?
The clinical buzzword is encephalopathy.
Encephalopathy.
Right. Multiple sclerosis, especially in its early stages, causes focal neurological deficits. An MS patient might suddenly lose vision in one eye or experience numbness in their left leg.
But their brain's overall processing power is fine.
Exactly. They're sitting in the ER, fully awake, fully alert, having a normal conversation with the doctor about their numb leg.
And ADEM is different.
Fundamentally different. The inflammation in ADEM is so globally severe, so widespread across the entire brain simultaneously, that the patient suffers profound encephalopathy, meaning an altered mental status.
What does that actually look like to the parents in the room?
It looks terrifying. The child is not just experiencing a weak arm. They are profoundly altered. They might be severely delirious, confused, unable to recognize their parents.
Oh my God.
They might experience drastic behavioral changes, extreme somnolence where they literally cannot be kept awake, or they may even slip entirely into a coma.
Just from the inflammation.
Yes. The actual gray matter and global processing networks of the brain are being suffocated by the intense inflammatory response in the surrounding white matter.
So if a test question or a real-life clinical scenario says, child develops severe confusion and multifocal neurological deficits 2 weeks after the measles, the severe confusion points directly away from MS and straight toward ADEM.
Exactly.
And you use that word again, multifocal. We talked about how chaotic the symptoms of PML were, but with ADEM, because the immune system is unleashing its entire arsenal at once, the scale of the damage seems almost incomprehensible.
The word disseminated in acute disseminated encephalomyelitis means exactly that. It is attacking everywhere at once.
The symptom list in the texts is staggering.
Because the immune SWAT team is pouring out of every vein in the central nervous system. They hit the corticospinal tracts, causing paralysis. They hit the spinothalamic tracts, wiping out the ability to feel pain or temperature. They hit the dorsal columns, taking away proprioception.
And the cerebellum?
They flood the cerebellum, causing severe ataxia and dysmetria, meaning the child cannot coordinate their movements to even reach for a cup of water.
And it hits the brainstem, too.
Right, damaging the cranial nerves, causing double vision, facial drooping, and the inability to swallow. And the riot often extends down the spinal cord, causing transverse myelitis, leading to a complete loss of bowel and bladder function.
All of those pathways failing simultaneously, you are basically watching the electrical grid of a massive house sparking, smoking, and shorting out in every single room at the exact same time.
All because the security guards are ripping the drywall apart to find a burglar that isn't even there.
And when you put that patient in an MRI scanner, the images reflect that massive violence, right?
Absolutely. Earlier, we said MS lesions look like small, discrete, punched-out ovals of white matter damage.
But in ADEM?
In ADEM, the MRI shows large, bilateral, cloudy, poorly marginated lesions.
Like clouds.
Exactly. They look like massive explosions of bright white inflammation scattered throughout the brain and brainstem. The blood-brain barrier is completely blown open, so if you give contrast dye, the lesions light up brilliantly.
So how do you stop a riot of this magnitude? I mean, you can't give antibiotics and you can't just wait it out because the swelling will literally crush the brain inside the skull.
Because the core problem is an overactive immune system, the emergency treatment involves chemical suppression. Doctors immediately administer high-dose intravenous corticosteroids, like methylprednisolone.
What do the steroids do?
They essentially flood the immune system with signals that tell the T cells to undergo apoptosis to self-destruct. And they rapidly force the blood-brain barrier to close back up, cutting off the supply of new inflammatory cells.
And if the steroids aren't enough to shut down the riot,
Then they escalate to IV or intravenous immunoglobulin, which is basically an infusion of healthy neutralizing antibodies that bind up the confused immune signals.
or plasmapheresis, right?
Yes. Essentially, hooking the patient up to a dialysis-like machine that physically filters all the patient's blood, scrubbing the attacking T cells and rogue antibodies out of circulation.
That is intense.
It is, but because ADEM is monophasic, if the critical care team can extinguish the fire and keep the patient alive through the acute swelling, the riot ends.
And they recover.
The child often makes a remarkable gradual recovery over several weeks as the oligodendrocytes slowly rebuild the myelin. Though, some patients are left with permanent neurological scars.
Okay. We've seen the viral assassins silently murdering the workers in PML. We've seen the friendly fire immune riot tearing the house apart in ADEM.
Yes.
But what if there is no virus? What if there is no immune attack? What if the threat to the myelin isn't an invader or a riot, but simply the water and salt balance of your own blood?
Now we are entering the territory of central pontine myelinolysis, commonly abbreviated as CPM.
Right, or osmotic demyelination syndrome, ODS.
Exactly. And from a physiological standpoint, this might be the most fascinating and biologically tragic mechanism we discussed today.
Because it's not a bug.
Right. The trigger for this massive destruction isn't a pathogen, it's physics.
This is the scenario I mentioned at the very beginning of the deep dive. The tragedy of the IV saline bag.
The rapid shift.
We are talking about the destruction of myelin, specifically in the central pons, which is this critical, densely packed structure in the middle of the brainstem caused by a rapid change in serum osmolality.
Right.
To understand how a bag of salt water can cause paralysis, we have to look at how incredibly adaptable the human brain is when faced with chronic problems.
We have to start with hyponatremia, which simply means low sodium in the blood.
Okay, so imagine a patient who develops hyponatremia very slowly over weeks or months.
This classically occurs in patients suffering from severe chronic alcoholism, chronic malnutrition, or certain long-term medication side effects.
So the sodium levels in their bloodstream slowly drop lower and lower. Now, the fundamental law of osmosis in biology is that water follows salt.
Water always wants to move across a membrane to dilute the side that has a higher concentration of solutes.
Right, so if the blood inside the blood vessels has very little salt, but the brain cells sitting right next to those vessels are packed with their normal, healthy amount of cellular salts and proteins, the blood is relatively dilute and the brain cells are relatively salty.
Yes.
The water in the blood looks at those brain cells and says, I need to go in there and balance things out.
Exactly. The water wants to aggressively rush into the cells of the central nervous system to dilute them.
But here is the critical anatomical problem. The brain is entirely trapped inside a rigid, unforgiving vault of bone called the skull.
Right, it can't expand.
If the billions of brain cells absorb a rush of water, they swell up. The brain expands, but it has nowhere to go.
This leads to cerebral edema. The pressure builds, the brainstem gets crushed against the bottom of the skull, a process called herniation, and the patient dies.
But that doesn't happen immediately in chronic hyponatremia.
No.
The patient survives for months with this low sodium. How does the brain prevent itself from swelling and crushing itself against the skull?
Through a brilliant, desperate metabolic adaptation.
How does it do it?
As the blood sodium slowly drops over days and weeks, the brain cells sense the osmotic pressure changing. To prevent the water from rushing in, the cells actively start throwing cargo overboard.
Cargo overboard?
Yeah, they purposefully shed particles called osmolytes from inside their cytoplasm into the bloodstream.
What are osmolytes?
They're organic molecules like taurine, glutamine, and myoinositol. By dumping these molecules, the inside of the brain cell becomes less concentrated, less salty.
Okay, so it's like a submarine that is sinking under heavy water pressure, so the crew starts ejecting the heavy ballast water to equalize with the ocean around them.
That's a perfect analogy.
The brain cells dump their osmolytes so that their internal concentration perfectly matches the low sodium concentration of the blood. The water no longer wants to rush in, the swelling is prevented.
The brain has adapted to a hostile environment. It is a physiological masterpiece of survival.
Nice.
But this brilliant adaptation is precisely what sets the stage for the tragedy.
Because eventually they go to the hospital.
Right. This chronically adapted patient eventually ends up in the emergency room, perhaps because the low sodium is finally making them confused or lethargic.
An emergency room physician draws blood, sees a critically low sodium level, say 110 milliequivalents per liter, when normal is around 140.
And the doctor naturally thinks, this is dangerously low. I need to fix this immediately.
And this is the fatal error. The doctor hooks up an IV of hypertonic saline or normal saline and runs it fast, rapidly pumping sodium back into the patient's bloodstream to get the numbers looking normal on the chart.
The fast correction.
What happens to those heavily adapted brain cells when the blood suddenly becomes intensely salty again?
This is the osmotic shock. The doctor has rapidly introduced sodium, making the extracellular fluid outside the brain cells hypertonic, very salty, very quickly.
But remember, the brain cells spent the last month dumping all their osmolytes.
They are running on a skeleton crew of solutes. They're highly dilute inside.
Suddenly, the environment outside is a hyper-concentrated salt bath.
So the laws of osmosis violently reverse.
Yes. The physics demand that water must rush out of the brain cells to dilute the sudden rush of salt in the blood.
And because the IV is running so fast, the water is rapidly sucked out of the intracellular space.
The brain cells just shrivel up.
Specifically, the oligodendrocytes, they severely and violently shrink.
Imagine you've been sitting in a pitch-black room for 3 days. Your eyes have completely adapted to the darkness. Your pupils are wide open, maximizing every photon.
Right.
If someone suddenly kicks open the door and shines a 10,000 lumen stadium floodlight directly into your eyes, you are blinded. You are in physical agony.
Because of the sudden change.
Exactly. It's not the light itself that caused the damage. Sunlight is perfectly normal. It's the speed of the change from dark to light that your biological hardware couldn't handle.
The brain had adapted to the low-sodium darkroom, and the doctor just flipped on the saline floodlight.
It was the speed of the correction that destroyed the self.
A fantastic analogy. The rapid cellular shrinkage causes massive mechanical shearing forces and intense metabolic injury.
The oligodendrocytes, the electricians, are literally crushed, dehydrated, and torn apart by this rapid shift in water volume.
As the cells die, they undergo apoptosis, and the myelin they were maintaining rapidly breaks down.
The USMLE texts provide a brilliant, haunting mnemonic to remember this devastating sequence of events. It is 5 L's: low, lose, lift, leave, lose.
Oh, that one, yeah.
First, the blood sodium is low chronically. Second, the brain loses osmolytes to adapt and survive. Third, a doctor lifts the blood sodium too rapidly with an IV.
Fourth, because of that rapid shift, water rapidly leaves the brain cells.
And fifth, the oligodendrocytes lose their myelin. Low, lose, lift, leave, lose.
It is a perfectly clear progression of a medical disaster.
But hold on, I need to push back on the anatomy here. The disease is historically called central pontine myelinolysis. Why the central pons?
That is the big question.
I mean, the entire brain is bathed in this changing blood chemistry. Every single cell in the central nervous system is experiencing the exact same osmotic shock. So why does the myelin in this one specific tiny region of the brainstem just absolutely fall apart?
It's a fascinating question that researchers have debated for decades. The source texts note that while osmotic demyelination syndrome can happen outside the pons, a condition called extrapontine myelinolysis, which can damage the basal ganglia or the thalamus, the central basis pontis is uniquely disproportionately vulnerable.
Okay, why?
And the prevailing theory ties directly to its highly specific architectural anatomy.
Like the structural engineering of that specific room in the house.
Exactly. The central pons is not just a uniform bowl of mush. It has a very unique, highly rigid, grid-like structure of heavily myelinated, intersecting white matter tracts.
Like a grid.
Yeah, you have massive descending motor fibers running vertically from the brain to the spinal cord. And intersecting them horizontally are dense bundles of transverse pontine fibers communicating with the cerebellum.
So it looks like a densely woven microscopic basket.
Right. Because of this tight, inflexible grid, the oligodendrocytes and surrounding support cells are packed in a way that severely restricts their physical ability to expand or contract.
Oh, wow. So when the rapid osmotic shift happens and the water is violently sucked out of the cells, they can't just gently deflate like a balloon in an open room.
They are trapped in this rigid grid.
The physical stress of the shrinkage in that specific matrix causes catastrophic mechanical shearing. The cells are essentially ripped apart by their own architectural confinement.
That is the leading pathological theory. The grid is too tight to accommodate the rapid fluid shift, so it snaps.
And the clinical outcome of this snapping is, it's truly the stuff of nightmares.
It is.
The texts point out that the symptoms are delayed. A patient gets their sodium corrected in the ER. The lab numbers look perfect. The doctor thinks they saved the patient.
The patient might even seem completely fine for 2 to 6 days.
The silent lag before the disaster.
Yes, because it takes a few days for the mechanically injured oligodendrocytes to fully die and for the myelin to dissolve.
But as the insulation rots away in the central pons, the power grid fails.
And because the pons contains the main descending motor tracts, the corticobulbar and corticospinal tracts, the failure is devastating.
What happens first?
The patient first develops dysarthria, which is slurred, unintelligible speech because the signals to the tongue and lips are failing. Then comes dysphagia, a total inability to swallow.
And then the damage reaches the main motor highways down to the limbs.
The corticospinal tracts demyelinate. The patient rapidly develops severe upper motor neuron weakness, progressing into quadriparesis paralysis of both arms and both legs.
And in the most severe, tragic cases of CPM, this progresses into what is known as locked-in syndrome.
The worst case scenario.
I want to walk through exactly what this means anatomically because it is horrific. The ventral pons, the front part of the brainstem, is completely destroyed by the demyelination.
Right.
This severs every single descending motor control wire going to the body. The patient becomes completely flaccidly paralyzed. They cannot move their arms, their legs, their face, their jaw.
They cannot speak, they cannot swallow.
They are entirely immobile.
But, and this is the defining horror of the syndrome, the sensory tracts running up the back of the spinal cord are often spared.
So they can still feel things.
Yes. And more importantly, the reticular activating system, which is a network of neurons located slightly higher up in the midbrain that controls wakefulness and consciousness, is completely untouched.
The cortex is fine.
The cerebral cortex, where all high-level thinking occurs, is completely fine.
Meaning the patient is fully awake, fully conscious.
They can hear the doctors talking, they can feel the bed sheets, they are cognitively perfect.
Yeah.
But they are entirely trapped inside a paralyzed shell.
Their entire biological house has collapsed, but the single light bulb in the attic is still burning brightly and nobody can see it.
The only way they can communicate is often by blinking or by moving their eyes straight up and down.
Why just up and down?
There is a very specific anatomical reason for that. The cranial nerves that control horizontal eye movements are located in the pons, so they are destroyed.
Okay.
But the cranial nerve that controls vertical eye movement, cranial nerve third, the oculomotor nerve originates higher up in the midbrain, above the zone of pontine destruction.
Wow.
It is literally the only motor wire left intact.
Communicating solely through vertical eye movements. When a radiologist looks for this on an MRI, the destruction is incredibly visually distinct, isn't it?
Yes. The text mentions a classic trident or batwing sign.
What does that look like?
If you take a cross-section MRI of the brainstem using those T2 or Fellair filters we talked about, the central pontine lesion glows bright white, and it takes the symmetrical three-pronged shape of a trident, perfectly outlining the destroyed grid.
It is a striking image. And the most heartbreaking reality of central pontine myelinolysis is that it is almost always iatrogenic, meaning it is caused directly by medical intervention.
It's an inflicted injury.
That is why the cardinal rule in internal medicine, drilled into every medical student, is the slow, highly controlled correction of chronic hyponatremia.
The rule is generally no more than 8 milliequivalents of sodium per liter in a 24-hour period.
You must raise the lights slowly to let the eyes adapt.
Because once the osmotic shock occurs and ODS is established, the medical texts clearly state there is no reliably effective therapy to reverse it.
The damage to the grid is done.
Prevention is the absolute only cure.
Wow. Okay. We have seen the viral assassination of the electricians in PML. We have seen the massive, confused immune riot in ADEM. We have seen the violent physics of rapid osmotic shock in CPM.
It's a lot.
Finally, we need to leave the brain entirely and move down into the long cables of the spinal cord. We need to see what happens when the insulation doesn't suffer a sudden attack, but instead undergoes a slow, creeping, systemic starvation. Let's talk about subacute combined degeneration, or SCD.
SCD is a progressive, relentless demyelinating disorder of the spinal cord.
But it's not a virus or an immune thing, right?
No. The sole culprit behind this disease is not an infection or an immune error. It is a severe, prolonged deficiency in a single molecule, vitamin B12.
Vitamin B12, also known as cobalamin. Now, when most people hear B12, they think of those tiny red energy pills at the pharmacy or like energy drinks.
Right, they think it's just for a boost.
But the biochemical reality of B12 is far more foundational. It isn't just about feeling energetic. It is a mandatory, essential metabolic building block required for DNA synthesis, red blood cell production, and crucially for this deep dive, normal lipid metabolism.
It is how your body processes fats.
The USMLE texts highlight the exact biochemical master pathways involved. Inside your cells, vitamin B12 acts as a mandatory cofactor for two highly specific enzymes, methionine synthase and methylmalonyl-CoA mutase.
Which, again, sounds like alphabet soup.
But without turning this into a chemistry lecture, what you need to know is that these enzymes are responsible for recycling and processing certain fatty acids.
And if you completely lack vitamin B12, these enzymes stop working. The metabolic assembly line grinds to a halt.
And when the assembly line stops, the raw materials pile up.
Exactly. Methylmalonic acid levels skyrocket inside the body. But more importantly, the body starts producing abnormal, unusual, odd-chain fatty acids.
Now think about what myelin actually is. Myelin is a lipid sheath. It is literally made out of concentrated fats.
So if the body's fat processing factory is broken, the oligodendrocyte electricians don't have the right building materials.
Right. They're forced to incorporate these abnormal, structurally unsound, odd-chain fatty acids into the myelin sheath they're building in the spinal cord.
It is like trying to build a brick wall using crumbling, misshapen clay instead of fired bricks.
The structure is inherently unstable.
Over a period of weeks to months, which is what the subacute, in the name subacute combined degeneration implies, this unstable myelin slowly degrades, frays, and breaks down.
The insulation literally rots away from the inside out because it was built with cheap, toxic materials. And the combined part of the name tells us exactly which walls of the house are rotting.
Right. It hits two specific pathways.
SCD classically and symmetrically destroys two major specific white matter tracts running through the spinal cord, the posterior columns and the lateral corticospinal tracts.
Let's map this out clearly.
Yeah, so everyone listening can physically feel what losing these tracts means.
Okay.
Let's start with the posterior columns. These are the thick bundles of myelinated wires running right up the back portion of your spinal cord.
They are responsible for transmitting three specific sensations to your brain: fine touch, vibration, and proprioception.
Proprioception is arguably the most vital of the three and the most devastating to lose. It is your body's internal subconscious GPS system.
It is how your brain constantly knows the exact position of your joints, your arms, and your legs in three-dimensional space without you ever having to look at them.
Like if you close your eyes right now and raise your left hand, you know exactly where your hand is. You can feel the angle of your elbow. That is proprioception.
So as those posterior columns slowly demyelinate and short out due to the B12 starvation, the electrical signals from the joints can't reach the brain.
The internal GPS completely fails.
The patient develops a condition called sensory ataxia. They become incredibly uncoordinated and clumsy.
But here is the fascinating distinction. They aren't uncoordinated because their muscles are weak. Their muscles are fine.
Exactly.
And they aren't uncoordinated because the balance center in their cerebellum is broken. They are uncoordinated because their brain is completely blind to where their feet are planting on the floor.
The brain is sending the command to walk, but it isn't receiving the confirmation receipt that the foot actually hit the ground.
Which leads to a very specific way of walking, right?
Yes. The analogy that vividly illustrates this is the concept of walking in the dark. The clinical texts describe these patients developing a very specific, wide-based, unsteady gait.
They sort of stomp, don't they?
They do. They often lift their legs abnormally high and literally stomp their feet down onto the floor. They are stomping in a desperate attempt to send a massive heavy impact signal up the remaining sensory wires.
Just begging the brain to acknowledge, okay, the foot has made contact with the floor.
But because the internal GPS is broken, the patient starts to rely entirely on their visual system to compensate. They constantly stare at their feet while they walk.
Their eyes are doing the job that the spinal cord used to do.
Exactly.
And this leads to a classic, highly testable clinical exam maneuver. If you take a patient with subacute combined degeneration and turn off the lights in the room, or simply ask them to close their eyes, they will literally fall over.
This is exactly what the Romberg test measures. The physician asks the patient to stand up straight, put their feet closely together, keep their arms at their sides, and simply close their eyes.
A healthy person might sway slightly, but their proprioception keeps them upright.
Right. But a patient with posterior column demyelination instantly sways dramatically and loses their balance the second their eyes close.
That is a positive Romberg sign.
It definitively proves that their balance was entirely dependent on their vision, compensating for the loss of proprioception. It points the doctor straight to the posterior columns.
But the disease is combined degeneration. It's not just a sensory problem. It attacks the motor system too.
Yes. The second major pathway destroyed by the abnormal fatty acids is the lateral corticospinal tract.
We've mentioned this tract before. It is the primary descending motor highway carrying voluntary movement signals from the brain cortex down to the body.
When the myelin here starves and breaks down, the patient develops classic upper motor neuron signs.
We should clarify what upper motor neuron means here. The nerves that actually touch your muscles are lower motor neurons.
Yes.
The wires coming from the brain down the spinal cord to tell the lower neurons what to do are the upper motor neurons. Usually the upper neurons send a constant inhibitory signal, kind of like a calming brake pedal, keeping your reflexes in check.
Precisely. So when the lateral corticospinal tract demyelinates, that calming brake pedal is disconnected.
And the reflexes just go wild.
The lower motor neurons become hyperexcitable. The patient develops hyperreflexia, meaning if a doctor taps their knee with a reflex hammer, the leg kicks out wildly.
They develop spasticity, which is a stiff, rigid increase in muscle tone.
And they exhibit a positive Babinski sign.
What's up?
Normally, if you firmly stroke the bottom of an adult's foot, their toes will curl downward. But in upper motor neuron lesion like SCD, stroking the foot causes the big toe to extend dramatically upward and the other toes to fan out.
Okay, the biology of this makes perfect sense, but I have to bring something up because I'm looking at a massive red warning flag in the USMLE text. It specifically calls this out as a high-yield exam trap.
Let's hear it.
Wait a minute. I thought vitamin B12 deficiency caused peripheral neuropathy. Numbness, tingling, the pins and needles feeling in the fingers and toes.
Right.
Isn't peripheral neuropathy a disease of the Schwann cells? Schwann cells are the entirely different type of cell that makes myelin in the peripheral nerves, out in the arms and legs, outside of the brain and spinal cord.
That's true.
So why are we talking about subacute combined degeneration in a deep dive dedicated to the failure of the central nervous system oligodendrocytes?
That is a brilliant catch, and it is the exact trap that board examiners use to trick medical students. You are absolutely correct in your underlying physiology. Vitamin B12 deficiency does indeed cause peripheral neuropathy.
Okay.
The abnormal fatty acids damage the Schwann cells in the hands and feet, causing that classic stocking glove numbness. However, the specific disease entity called subacute combined degeneration is, strictly by definition, a central nervous system pathology.
The text gives a simple rule to remember this. Where determines which myelin cell?
Yes, the location dictates the cell type. The pons is inside the central nervous system vault, so central pontine myelinolysis is an oligodendrocyte disease.
And the spinal cord.
The spinal cord is the main highway of the central nervous system. Therefore, the posterior columns and the corticospinal tracts inside the spinal cord are myelinated entirely by oligodendrocytes.
So SCD is the failure of the central oligodendrocytes, while the neuropathy is the failure of the peripheral Schwann cells.
Exactly. Subacute combined degeneration is the specific failure of those CNS oligodendrocytes to maintain myelin due to metabolic starvation. The peripheral neuropathy you mentioned is a completely separate manifestation of the exact same nutritional deficiency.
That makes perfect sense. The whole biological house is out of building supplies, but SCD specifically refers to the internal load-bearing walls of the spinal cord crumbling.
And radiologists can actually see this crumbling on an MRI. It creates a very classic visual.
It's an inverted V sign, right?
Yes. If you take an axial cross-section image of the cervical spinal cord, looking down the tube of the spine from the top, the demyelination in the two posterior columns at the back of the cord glows bright white on a T2 MRI.
It looks exactly like an upside-down letter V.
It's a very striking radiologic image, and the critical clinical point here is intervention.
Which is pretty straightforward, right?
If a doctor recognizes the sensory ataxia, the positive Romberg, the spasticity, and confirms the low B12, they can treat the patient with massive intramuscular injections of vitamin B12.
Just give them the supplies.
If caught early enough, flooding the system with the missing cofactor restarts the metabolic assembly line.
The abnormal fatty acids are cleared out.
Healthy lipids are produced, the oligodendrocytes repair the myelin, and the symptoms can actually be halted and even reversed. But, just like in PML, if the demyelination is allowed to persist for too long, the raw, uninsulated copper axons in the spinal cord will eventually undergo Wallerian degeneration.
They will die.
And once the axon itself is dead, no amount of vitamin B12 will bring it back.